Hello everyone, I drew the long straw and I get to go first. So yes, talking about genitourinary tissue. So as you'll see in a few minutes, I haven't got much cancer research done yet, but plan to get started on that soon. So the aspect of genitourinary tissue that we work on is the testes, and it's related to male infertility. So there are worrying trends in fertility overall across the globe, whereby by the end of the century, the average age will have increased from about 30 to about 50, and this has a lot of knock-on effects for society in terms of who's supporting who, have we got enough young people to support an ageing population. So we should be doing everything we can to encourage people to procreate, to maintain a healthy fertility level, but we are not doing that because infertility is still a rising issue. It affects 1 in 6 couples, and maybe not known to everyone is that 50% of cases are due to a male factor. And to link this into cancer, then a lot of childhood cancer survivors are involved in this male factor infertility, because the chemotherapeutic treatments that they receive are germ cell arresting, so they do experience, a lot of them, fertility issues in later life. And you can't, with female childhood cancer patients, you can freeze reproductive cells. In males, you cannot. So this issue of male infertility is also compounded by the fact that overall sperm counts have been decreasing for the past 50 years, and there's a 52% decline in sperm concentration. And if you do project this line downwards, it reaches 0 by the end of the century. You should never do that if you're writing a scientific paper, but if you're trying to get money, it's very useful. And worryingly, combining these three with the fact that there is no effective treatment for male infertility. There are surgical options where you can go foraging for sperm cells in people who have no sperm in their ejaculate, but other than that, there's no kind of effective pharmaceutical or hormonal treatments. So what we want to do is create preclinical models to help evaluate treatments and see if there's anything that we can do to help these people. So when you talk about the anatomy of the testes and you talk about creating a preclinical model, you'll notice that it is quite complex. So you have these seminiferous tubules embedded in interstitial space, and it's inside in these tubules that the actual sperm production takes place. So not only is it very complex anatomically, it's also very complex physiologically. So the germ cells go through a number of stages of meiosis and mitosis before they reach the tadpole-like shape that you're more familiar with. So all to say here is that male fertility is an issue, and we do need preclinical models, and that preclinical models created of this organ is quite challenging due to the complexity of it. So what we do in our group is we work with the native tissue, trying to characterize it, and we also perform modeling of that tissue. So that's physical modeling of the tissue. We create physical models of the tissue. So to kind of get myself out of jail here, you could apply this to cancer tissue, and we do intend to apply this to cancer tissue as soon as we get cancer tissue. So what we do get is biopsies, blood, paraffin blocks, and what we do with this tissue is we characterize the physical properties, the chemical composition, the cellular expression, and then on the modeling side, we extract and sort cells, we extract out material from the tissue, and then we recombine that into a model system where we can fabricate things at high throughput. And the final step then is kind of comparing how this model tissue compares to the native tissue. So first things first is you need to actually have access to the tissue. So that's what I want to talk about next. And we have several kind of cohorts that we look at in the group. So we have fetal tissue, prepubertal tissue, which is taken from childhood cancer patients who are about to undergo fertility arresting treatment. They will preserve some of their testicular tissue in the hope that later there will be a platform to mature the cells in vitro into mature germ cells. Currently, one does not exist. We then have adult fertile patients and adult infertile patients, and we get these tissue samples from a range of collaborators. So we have gender reassignment surgeon Jochen Hess in Essen, Germany. So we get whole testes from him. From the Blackrock Clinic in Dublin, from Mr. Ivor Cullen, we get a small piece of testicular tissue every time that a patient goes for a surgical fertility treatment where they're trying to actually extract sperm from the testes. We get a little bit of that. We piggyback on existing ethics to get the fetal and prepubertal tissue from Edinburgh and from the Caroline Institute, and then we're in the closing stages of securing some testicular cancer tissue from Niamh Peters in UHL. So I think it's important first to show the actual anatomy then. So in all these cases, what you're looking to see is there complete spermatogenesis. Is the tissue actually producing sperm, or have you got arrested spermatogenesis? So here we can see complete spermatogenesis. This is a Mason's trichrome stain where you can see collagen is in blue and cells are in red. And on the H&E stain, you can actually see mature sperm cells here being produced inside the tubules. But then you have the case where there's arrested spermatogenesis. So this is a man experiencing infertility. You can see that the tubules are collapsed in on themselves. The basement membrane in blue is completely thickened, and there's complete loss of function here in terms of producing sperm cells. So, this is what it kind of looks like in terms of collagen and cells. This is what it looks like in terms of these. We have myoid cells which surround the tubules, and they're like smooth muscle cells. They provide contraction and they push the sperm cells through. And your Sertoli cells are like somatic cells that line it and support the germ cells in coming through and maturing. But you can see the difference here across fertile and infertile. You can see that there's concentric rings of smooth muscle or myoid cells here. There's no concentric rings here, and all of these cells have collapsed into themselves. So just to kind of give you an idea of what the morphology of the tissue is, because I know not everyone will have seen the inside of a testicle before. So to take you on to the next point, I'm just going to take you through a few of these points. We're currently working on all of these. We have different members of the team working on different aspects of the tissue. But the first thing I want to talk about is characterising the physical properties. So Isabel is performing this work here. All of these pictures are taken from fancy dress at our annual conference. So we wanted to characterise the mechanical properties, because the mechanical properties are one of the factors which would influence cell maturation. You have many, but extracellular matrix mechanical cues are one of them. There have been lots of studies to show this, particularly looking at the matrix viscoelastic properties. So the viscoelastic properties, you'll all be probably familiar with the concept of stiffness. Stiffness is the instantaneous response of a tissue to a deformation, whereas viscoelasticity is a time-dependent response of a tissue to a deformation. So that's what we're trying to characterize here. And we use what's called a nano-indenter. So we have a probe that indents the tissue, and we use laser interferometry then to see how much this cantilever beam has displaced, and we're able to relate that then to displacement of the tissue and also measure the force that it creates. So you'll see here is a tubule. It's being indented here by our probe. And what this gives you then is this kind of a profile here, a load indentation graph. And we'll indent the tissue, and then we'll also wiggle the probe as well. And this kind of allows us to understand how that tissue responds to immediate kind of, it's called storage modulus and loss modulus, how the tissue stores energy and how it loses energy. So those are kind of the metrics that we carry forward then. So we did this first in our healthy patients. So you can see here's the H&E staining. The tubules are nice and open. The basement membrane is nice and thin. And here's a bright field image of where we're indenting. So you can, because the size of this probe is 50 nanometers, you can get a lot of indentations. So we're looking here at about 60 indentations. This is Young's modulus. So this is a metric that you'll probably be a bit more familiar with. This is just the stiffness of the tissue. It's instantaneous response to deformation. But we can also get storage modulus and loss modulus. And just the important point to note here is that we're getting a Young's modulus or stiffness of around 1,000 pascals, so 1 kilopascal. And we're able to look at the seminiferous tubules, we're able to look at the interstitial space in between them. So what can we do with this information? Well, the first thing we can do with this information is compare to the kind of substrates that we grow these cells on in 2D. So here's polystyrene up here at around the megapascal range anyway, definitely, or gigapascal range, which is stiffer than bone, stiffer than skin, stiffer than ocular tissue. And where does the testes place? Down here at the bottom. So growing testicular cells on polystyrene tissue culture plastic is not a very good representation of the microenvironment mechanically that they experience in vivo. Probably more interestingly, we're able to compare our healthy fertile tissue to our infertile tissue. So this is the infertile sample here. We did a number of indents again. And what we were able to see here in the fertile versus infertile, so fertile in purple, arrested in blue, you're able to see there's a marked difference in terms of the mechanical properties. So what we want to do next is apply these techniques to our entire cohort. So we've done it for some fetal tissue. We want to do it for prepubertal and do it across a wide range of fertile and infertile, just to understand the mechanical differences that exist in the environment that these cells reside in. And we want to be able to then recapitulate that in our preclinical model. So the next thing we want to talk about, I'm just going to jump around here, whatever took my fancy last night as I was putting these together. In terms of how do we fabricate a model then, like, how do we actually go from input from this side to actually making a model of this. So here's Jishnu, also in fancy dress, co-supervised by Tara Dalton in the School of Engineering. So we're using sodium alginate as our model material here to help to act as the basis for our preclinical model. So what Jishnu will do is he'll take sodium alginate, cross-link it with a calcium EDTA solution, and drop it into a catalyst then, so that we get these little microspheres of sodium alginate. And he's done a great sweep of all the different properties or all the different concentrations. So these are our stable hydrogels here. These are unstable hydrogels. They don't maintain their shape at all. And when we compare the mechanical properties of our sodium alginate material to those of the native material, we can see this line here represents the sodium alginate. We're getting a good approximation of the mechanical environment that the cells reside within using this material. So Jishnu has then gone on to culture primary testicular cells inside in the sodium alginate. So here you can see one of these microgels here, and you can see the cells. This is just we were having stained here for live dead. Well, live, we didn't have any dead, so we don't know what's dead inside, but this is after 1 day of culture. A lot of this is hot off the presses, guys, so I don't have a lot of conclusive evidence here to suggest anything, but just kind of showing you kind of the methods that we use and kind of the approaches that we're developing. So we were very happy with this. You can get your cells alive inside in your sodium alginate hydrogel, but we want to be able to do this at high throughput. So we're using microfluidics to create hundreds of these small microgels at high throughput, using microfluidic systems, and we're able to create these droplets due to the shearing forces that occur at a T-junction like this. So, as I said, hot off the presses, this is us creating our preclinical models at high throughput. The camera autofocuses, we don't know how to stop it. But just to showcase that we're capable of creating these preclinical models that are mechanically representative, and we're able to do this at high throughput. So we also, there's Nazrin there, also in fancy dress. Thanks, Nazrin. In order to, like, sodium alginate is derived from seaweed. It's not a very chemically representative kind of material for preclinical models. So we wanted to use the native extracellular matrix. We get a lot of these samples. We get a lot of tissue from collaborators. So what we do is we take the native tissue, we decellularize it, we take out the native ECM, and we confirm via staining that we've removed all of the cellular content, and what we're left with is just the tissue. So here in the pentachrome stain, I don't know if anyone's ever used this before, very hard to get working, but it stains collagen for yellow, mucins for blue, muscle for red, and the cells are then kind of a purple. So we were able to show that we were taking out all of the cells, and also upon DNA quantification, we were able to see that we're reducing the DNA content in there. So we want as low a DNA content as possible. We don't want an immunogenic response from the cells that we're trying to culture. We're trying to create an environment that represents the native environment by using actual native tissue. And we are able to form hydrogels from this material here. So here we see it just native, just the ECM used by itself. But here we can see we've added some cross-linking agents. We're trying to get tailoring the mechanical properties of it. So that's all I wanted to talk about today. Maybe if you have me back next time, I'll talk about some of the other stuff that we've going on. But just kind of more to show the approach that we're using. And this can be tissue agnostic as well. Any tissue that we get in, we're able to characterize the physical properties, chemical composition, cellular expression, and also extract out cells and sort them, extract materials and fabricate them on models. So, yeah, if anyone wants to discuss any of this afterwards, I'm very happy to do so. And just to acknowledge our funders, the ERC, the UL Foundation, the LDCRC, Alwyn got the Fulbright and the Marie Curie, SFI and IRC now as well, and to acknowledge the testies besties who actually do all the work here. So 8 current members, including myself, and 4 former members. This is us in our complete fancy dress there at various conferences. So yeah, it's been good time so far. So thanks for that, guys.
Usually most people can hear me. With the increasing infertility, are you also seeing an increasing rate of testicular cancer
yes,
in those same groups?
Yeah, Yeah, so when you talk to the surgeons and you read the literature, there's this hypothesis of a bad testicle. So, it experiences infertility, it doesn't produce sperm. It's also more likely to undergo, cancer development. So there can be a number of triggers here, like if you have an undescended testicle, it has to be manually descended within 1 year of birth, and if that doesn't happen, the environment completely changes. So kind of like the risk factors for infertility, or the indicators for infertility and cancer are very similar. Small, hard testicles that aren't producing any sperm are very likely to undergo transformation into cancerous as well. So, I mean, one of the less probably worrying ones, because you can just take it out. But also, we kind of need, if you do take it out, we need systems to develop mature sperm for those patients as well, because they're left with no options after that then.
Brilliant. Kieran, one quick one.
Just a quick one. I'm very interested in the thickening basement membrane that you saw.
Yeah.
Has that been characterized, the ECM components?
So we're trying to characterize ECM components, right? We are also very interested in that, all right. We're characterizing, we've looked, and there's a lot of collagen 1 in and around there, and so like there's this, crosstalk between the Leydig cells in there that produce the hormones that trigger a lot of the processes in the seminiferous tubules, and if they're blocked by collagen 1, this kind of collagen 1 deposition, is it cause or effect? We don't know. Is it the mechanical environment as well? Because instead of deposition, is it an unraveling? It kind of looks like there's a thinning of it in addition to an increased presence of it. So, yeah, we are looking at collagens and laminins, fibronectins. We're doing specific stains for those as well, because we're very interested in that as well. Yeah.
All right,
Talk to you after. Sounds
Yeah.
good.
Cool.
Move on. Right, thank you
Cool.
very much.
Thanks, guys.
Now we have Michael Lynch.
Hi everyone. For those I haven't met before, I'm Michael Lynch. I'm a PhD student in the Colorectal lab in the School of Medicine. And today I'll be talking about some of our work on single-cell analysis of the CCRCC tumour microenvironment. So clear cell renal cell carcinoma is the most common subtype of renal cell carcinoma, at about 70%. It's more common in men, and men also have a worse prognosis, with an average age of diagnosis at about 60 years. So it's usually brought on by loss of function in the VHL tumour suppressor gene, with other risk factors including obesity, smoking, and high blood pressure. So it usually metastasizes to either the lung or the bone, and this is associated with reduced overall survival. So CCRCC has quite a unique microenvironment, and it's often used as a model for translating immunotherapies to solid tumours. So it was initially considered quite immunogenic, meaning it elicits an immune response. But paradoxically, even though it has quite a high level of CD8 T cell infiltration, this is associated with a poor prognosis in CCRCC. So some recent single cell studies have shown that depending on the immune cells present in the microenvironment, these can either suppress or paradoxically promote the actual tumour growth. It's also a very heterogeneous tumour, meaning there's large differences within and between different patients. So in collaboration with our collaborator, Dr. Yufei Wang at the Mara School Lab at Dana-Farber, we're in the process of trying to characterise differences between the primary RCC tumour and the lung mets, as well as linking that with clinical response. So we collected samples from primary RCC as well as lung mets, including one lymph node. We performed single-cell sequencing on the TEX platform and are now in the process of analysing the data. So this includes removing low-quality cells, demultiplexing, dimension reduction and clustering to kind of group similar cell types together, and then differential gene expression, which will then tell us which genes are being expressed that are differentiating these clusters. So single-cell sequencing is really useful because it allows us to look at cell types at a really granular level and in an unbiased manner. So most importantly, this means we don't really need to know what the markers we're interested in are before we start our experiment. We're sequencing the full transcriptome, and so this allows us to give us this level of detail. So people who work with single-cell data will be quite familiar with these kind of t-SNE plots. For those of you who aren't, we usually start off with somewhere in the order of 30,000 genes We'll perform a PCA, or principal components analysis, to compress this down into 50 to 100 different dimensions, which express most of the variability in the data. The t-SNE plot, or also you'll see UMAP plots like we have here, then further compress that in a nonlinear manner into a 2D embedding so we can visualize the data. So each dot here represents a single cell, and we'd expect cells that are quite similar in terms of their gene expression to be closer together on the graph. So as you can see here in our data set, we've been able to annotate different immune subpopulations like CD8 and CD4 T cells, myeloid cells, B and NK cells. And in the non-immune compartment, we have the CCRCC cells, stroma, progenitor, and epithelial cells. So by using single-cell sequencing, we can identify rare populations, for example, T follicular helper cells, as well as different states of similar cell types. So for example, here, we've 2 distinct CD8 effector populations, as well as a third CD8 cluster, which has an exhausted phenotype. So first, comparing the compositions between primary and metastatic, overall we see they're very similar. There's very few unique cell populations to either one or the other. If you look closely, you'll see these 2 populations here, one of which is a T cell cluster with very high IFID expression, the other is an NK cluster. But these are associated with singular patients rather than a consistent primary Mets response. And this brings us back to the large level of heterogeneity we see in CCRCC. So we're currently in the process of carrying out differential gene expression within each cluster to determine if there's more subtle transcriptomic differences between the primary and the Mets microenvironment. For the lymph node then, that's kind of doing its own thing a little bit, which is somewhat to be expected, with very high levels of CD8 exhausted cells, CD4 memory, and B cells. So compared to bulk sequencing methods, single cell allows us to identify some rare cell populations which have potentially important roles in either response or prognosis for patients. So on the left here, we're comparing the abundance of 2 cell types, B plasma cells and T follicular helper cells, which are associated in the formation of tertiary lymphoid structures, which may play an important role in patient response. So in terms of the primary on the left, we can see very low abundance of both of these. On the Mets then, we've really got a large amount of variability. So for example, we can see sample 9 and sample 11 have considerably higher proportions, and the remainder of the Mets are quite low. Again, coming back to this heterogeneity. So if we plot the correlation, we usually see where there's high T follicular helper cells, we also have high T plasma cells. If we then overlay some data we have on the clinical outcome of these patients, so their ongoing overall survival, we see that these 2 Met samples up in the top right corner actually have quite good clinical outcomes of over 5 years ongoing survival, which is quite good for metastatic patients. So this raises the question then, that we're currently trying to figure out, so is this indicative of a response or is it due to another factor? For example, both these patients also had neoadjuvant therapy, which may have altered the microenvironment. And that's something we're working on at the moment. So single cell experiments are typically much more expensive than bulk experiments. So one of the techniques used to address this cost is multiplexing. So instead of sequencing cells from each sample, 1 sample at a time, we pool the samples together and sequence them on the same lane. So to figure out which cell came from which sample, we stain each sample, or each cell from each sample, with a unique antibody, which we can then link back to the sample. However, for anyone who's worked with antibody staining before, we'll get this non-specific binding. And so if we have a lot of non-specific binding or a large overlap in the bimodal distribution, it's pretty hard to actually work out what's signal and what's background. So we developed a method, DMUX-SNP, which looks at its hashing data and figures out, okay, these are the cells we can call with really high confidence. So we'll use those to train a classifier for any cells that are either uncertain or can't be called due to low hashing quality. We then look into the SNP space. So SNPs are single nucleotide polymorphisms. So for example, on the bottom here, we have this uncertain cell. It's clearly surrounded by hashtag 2s in SNP space. So we can call it a hashtag 2. This is packaged in the DMUX-SNP bioconductor package, which is available. We also have a preprint describing the method on bioRxiv. So to summarize, we can use single cell sequencing we can form an unbiased and high resolution and a hypothesis generation in complex tissues. And as well as that, we can look at the gene expression of rare cell types and potentially link these to clinical outcomes. On the computational method side, we can integrate with existing or public data sets to increase our statistical power. And from our in-house methods development, we can recover negative cells to improve the overall cost effectiveness of single cell sequencing. Happy to take a question or two now, or pass on to whoever's next. Thank you.
Do we have any questions? Kieran?
I'm really interested in this and going through your package.
Yes.
Your DMF snip. I'm just wondering, this is purely self-serving, but from you've built it against the whole transcriptome data.
Yep.
Is it deployable on targeted data?
So in its current form, I'm going to say no. We're reliant on a certain number of genes being expressed across most of the cells in the data set, which I think will be unlikely to have in a targeted panel because you're unlikely to be interested in genes that are expressed everywhere. It might be adaptable in some form, but based on its current form, I'm going to say no. But again, I presume if you have an experiment in mind for that, it's something we can take a look at.
All right. Thank you very much.
Cool, thank you.
And now we're going to have one of my colleagues in the biological sciences, Jason Bennett, going to talk about some of his research.
Thanks, James. So just a warning, this is only 4 months old in terms of a research stream, so it's very, very preliminary. So it's more of a story. So basically, my background in research is basically looking at deciphering the roles of NF-kappa B in kind of integrating anti-apoptotic and immunosuppressive signaling in cancer. So the NF-kappa B family of inducible transcription factors, through the induction of a vast array of genes, act as key regulators of development, immunity, inflammation, cell survival, and defense responses. However, basically, they also play a role. They contribute to, the pathogenesis of most of the chief threats to human health. And as I said, it's abnormal NF-kappa B activation is a key hallmark of cancer, and that's where I was kind of spending the last 10 years, but this is a new stream where I'm looking at endometriosis. So in terms of NF-kappa B and cancer, it was basically the drive oncogenesis in terms of induction of genes that suppress apoptosis and orchestrate inflammation in the TMEs. And now I'm kind of looking at what's happening in terms of endometriosis. So endometriosis is a chronic inflammatory hormone-dependent condition characterized by the presence and growth of endometrial-like tissue outside the uterus. Okay, it affects about 10% of women of reproductive age. Okay, extrapolating about 190 million women, but this is a very conservative estimate because of the gaps in terms of knowledge about the disease, diagnosing, etc. So that is probably much higher. These endometriotic lesions basically occur mostly in the lining of the pelvic cavity. So basically, they're subdivided into superficial peritoneal lesions, makes up about 80% of all diagnosed cases. Ovarian endometriomas, these kind of chocolate cysts you see here on the ovaries, and deep infiltrating endometriosis. These are lesions that basically penetrate that tissue to a greater extent of 5 millimeters. But it also can occur in extrapelvic regions, such as the upper abdominal visceral organs, etc., and it can even basically be found in the thoracic cavity. Okay. In terms of what we don't know, in terms of the origins of endometriosis, it's a complete enigma. Okay, still no one really knows exactly what's happening. Obviously, because of the lack of research, the lack of models that basically mimic the disease. So we have a series of theories. Okay, so Sampson's theory was first postulated, it's coming up to nearly 100 years since it was basically first published. And this is the theory of retrograde menstruation. Basically, you get an efflux of tissue via the fallopian tubes out into that cavity, okay, out into the pelvic cavity. And but the problem is this efflux occurs at about 90% of healthy females. So basically, there's no matching in terms of basically how many people have endometriosis. So there's some other component, either genetic or most probably some sort of immune dysfunction that basically facilitates lesion implantation and growth. The other theories, you have colonomic metaplasia theory. This is basically where basically it's involved in a defect in terms of the development of the reproductive tract, whereby metaplasic differentiation of the colonomic epithelium into endometrial cells that basically seed the pelvic cavity. And then you have basically the stem cell progenitor cell theory, where basically neonatal bleeding can occur upon birth, and basically stem cells are seeded into the pelvic cavity and they remain dormant until adolescence. Okay, so, but these theories, again, are just theories. There's nothing concrete yet. In terms of the symptoms of endometriosis, basically pelvic pain, about 90% of pelvic pain is due to endometriosis, and then infertility. These are the key symptoms. But basically pain is kind of across the board in terms of the digestive issues, basically problems in terms of urinating, and it has a severe impact on all aspects of life. And there is now studies being ongoing in America where they're looking at the socio-economic impact of endometriosis, and it's running into the billions every year, whereby basically the problems in terms of diagnosis, treating over many years, and then basically the lack of productivity because of this painful condition. So as I said, diagnosis is an issue. You only get a definitive diagnosis upon laparoscopy, and basically the average diagnosis delay is about 8 years, which is a bit crazy. So why? Because of non-specific symptoms, no specific biomarkers, lack of awareness in terms of clinicians, etc., and the patients themselves, and then the stigma. This is the classic suffering in silence condition. That basically people will take it as a given. Okay. You come to treatments. What are the treatment options? You're just looking at the symptoms, treating the symptoms, painkillers, anti-inflammatories, hormonal treatments, where basically are built around suppressing basically the ovary. Okay? And if you're trying to conceive, that's a bit of a crazy tact to do. Okay? And then you have surgery, where basically you have 50% recurrence rate. So the therapies, the treatments are nowhere near where they should be. I was up in the charity, the Endometriosis Association Information Day, and it was basically people telling horrific stories about what they're going through, and then the treatment options were basically physiotherapy or acupuncture, which is just not where we should be. So there's a big lack of diagnostic imaging, non-invasive biomarkers, but there's no targeted therapies. We should be trying to clear these lesions from the pelvic cavity. So this is where NF-kappa B comes in. So basically, NF-kappa B is held in active state in the cytoplasm. You get stimuli activating the IKK complex, basically phosphorylates, and you end up with eubiquitation of the I-kappa B alpha protein, which basically gets basically destroyed by the proteasome, and then NF-kappa B is free to basically enter into the nucleus and activate all its different gene targets. Okay? But the problem is that because it's basically there's a really drive to basically target this pathway in terms of malignant and non-malignant diseases. People came up with these different inhibitors. But basically, because if you try to inhibit the main components of NF-kappa B, basically because of the severe on-target basically side effects of globally suppressing NF-kappa B, you're getting nowhere. No NF-kappa B inhibitor has entered the clinic. Because of basically the seminal paper by Kieran's group in America, where basically when they developed this IKK inhibitor, they treated some mice with it, basically because of an unknown role of NF-kappa B in terms of IL-1 beta processing. Basically, the mice got overt systemic inflammation and just dropped dead. And that killed basically the field for a number of years. So in a previous life, I was basically a postdoc working in this field, and our theory was that basically if NF-kappa B functions by targeting or basically inducting genes, can we identify and target the downstream effectors of that oncogenic process, the genes that drive cell survival and drive inflammation in the tumour. So we identified a protein, GAD45-beta, that basically functions by blocking the junk apoptosis pathway. So basically GAD interacts, it binds, it blocks basically the ATV binding site of MKK7. And we looked at this in terms of multiple myeloma. So this basically is the classic paradigm for NF-kappa B driven cancers. So basically it's an incurable malignancy of these plasma cells. About 20% of all primary multiple myeloma cells, and the majority of multiple myeloma cell lines display aberrant NF-kappa B activation. So when we silence GAD45 beta in multiple myeloma cell line U226, basically it undergoes apoptosis via activation of the JNK pathway. Okay, so this was kind of the first step. So we had this protein-protein interaction. Okay, so we were able to come up with basically a strategy to develop GAD45 beta inhibitors. So we screened a smart peptide library, a combinatorial library of about 20,000 tetrapeptides for peptides that were able to block the interaction of GAD45 beta and MKK7, which led to the development, after you go through all the chemical optimization steps, of a three amino acid DTP3 that went through all the preclinical screening, and eventually it's in the clinic. And basically, DTP3 was shown to induce, again, when you treat your multiple myeloma cell lines, was shown to induce cell death, apoptosis, again, through the JNK activation pathway. And as I said, it went into this proof of concept phase 1 trial. So basically, DTP3 was shown to have a clinical capacity to trigger a cancer-specific pharmacodynamic response by inducing JNK-driven apoptosis in multiple myeloma. And crucially, it was just targeting the multiple myeloma cells. It was leaving just the normal cells, had no effect when we ran it on the flow. So that basically was the design. By targeting the downstream effectors, you could potentially block NF-kappa B function. And this is kind of what I want to bring to endometriosis, because this is what the arena I've been kind of working on for the last 10 years. And if we look at endometriosis, when endometriosis became a factor in my life, I started to read up about it. And the more I read, the more I realized there were some similarities here. You know, inflammation is potentially a key driver for that initial initiation of endometriosis, implantation and progression. You know, the immune system, macrophages, should be able to basically clear those endometriotic cells when they're shed, so there's some sort of immune dysfunction. You know, angiogenesis, invasion, metastasis are also shared characteristics, and then you get resistance to cell death. The cells are surviving in this pelvic cavity where they shouldn't be, okay? And there has to be, in terms of that local microenvironment, there has to be some sort of rewiring of the cellular metabolism. And then the one that kind of threw everyone off there, published last year in New England Journal of Medicine, was basically that they identified in deep infiltrating endometriosis, they found some basically drivers, some somatic mutations of cancer driver genes, so basically in a condition that had zero evidence of transformation, nearly zero chance of actually being transformed. So my simple brain basically thinking, is there some sort of common abnormally regulated signaling pathways in terms of endometriosis initiation and progression? And lo and behold, we're back to endometriosis, and NF-kappa B. So NF-kappa B is constitutively activated in peritoneal endometriosis. So basically, can we identify now basically non-redundant effectors of the NF-kappa B pro-survival function in endometriosis? Mahdi is doing some work on this for me. She's looking at the 12Z human endometriotic epithelial cell line, and there is some sensitivity to NF-kappa B inhibition. So basically, they are sensitive to basically blocking the NF-kappa B pathway. Unfortunately, to get rich quick and retire early is probably out the window. GAD is not involved in basically this survival function. But we're going to basically silence REL-A, silence NF-kappa B in these cells now, and do what's known as a death trap screen. So basically, we can generate a cDNA expression library from TNF-alpha treated wild type 12Zs, basically transfect out those basically silenced cells with this library, induce cell death, and then hopefully extract some episomal DNA. And we can repeat this cycle to basically, this is an unbiased method, and it should allow us to select genes with anti-apoptotic function, which facilitates the isolation of unknown genes. And then on the side, we're going to do some examinations of the immune dysfunction part of endometriosis, because basically a lot of the studies done are all very observational, looking at the peritoneal fluid. Basically, neutrophils are up, macrophages are up when you compare them to control patients. And Stephen, my PhD, is starting to do some work on 3D mixed culture spheroid models. As Eoghan says, hot off the press, this was actually done last Friday, because there was a bit of a panic to come up with something for the talk. But basically, we have our stromal and endometrial cells with their bioflow plates making lovely spheroids. But when you put THB1 macrophages in the mix, something is happening. It's the results I like to have where you actually see something happening. So Stephen's going to come along do the viability assays hopefully next week, and then start to look at the transcriptome, proteome analysis and see what's happening to these macrophages when they're in that mixed culture. But ultimately, I went to the BSI last November, and basically, if you're not in the tissue, no one wants to talk to you anymore. So basically, I want to lean on the key core texts in the LDCRC to get some endometriosis tissue with. I have talked to people in Limerick Hospital already, that shouldn't be a problem, and then do some single cell RNA-seq, and then do spatial phenotyping to see actually what is in these tissues, because no one has done any work on this. Okay? There was a very small single cell RNA-seq study in endometriomas published last year in Nature Genetics, so it's very novel. I've talked to the guys in Akaya, very novel. So hopefully this will be the start of a long research career, hopefully, if it doesn't fall apart. So thanks very much, and thanks to Mahdi. Softball, James.
Hi.
Hi.
Christina, I'm a colorectal surgeon. So
Yeah.
I just really want to thank you for this research. So I would do a lot of operating on deep infiltrative endometriosis with my gynecology colleagues, because this often affects the rectum as well.
The lesions and
Exactly.
the adhesions.
Exactly,
Yeah.
yeah. And often actually it's embedded,
Yeah.
and when we sometimes have to do resections, this is a population of patients that have been savagely underserved by our health service.
Yeah.
So I think for you and your research group and our region to lead on this is something really powerful for women's health.
Yeah.
And you mentioned like the impact of this, it's fertility, it's chronic pain, and it's opioid addiction. And we're seeing more and more of that, and it's something we haven't really learned to address.
I only got into it because it affected my own family, basically.
Ms. Katie Field and Professor Osman Mahmood do a lot of operating
Yeah,
on these patients. So from the point of view
met-
of tissue samples, I'm
Yeah.
sure they would be more
Yeah.
than happy to collaborate.
There's they're the main base down in Limerick, and then there's a new clinic being set up in Dublin as well. So the networks are small, which might be an advantage, but hopefully we'll get some tissue this summer.
Thank you.
No problem. Thanks very much.
Thanks a lot.
Thanks, Dave.
Now for some diversity, we're getting Sinead's going to talk about some of her research.
Okay, thank you. So my research looks at developing optic fibre sensors for radiotherapy dosimetry. And I'm going to focus today on just one particular project that we have running. So radiotherapy is delivered in more than 50% of cancer. It's okay now. Here. So radiotherapy is delivered in more than 50% of cancer patients. And the aim of radiotherapy is to deliver the ionizing radiation to the tumour, maximise the amount of radiation you're delivering to the tumour while minimising the amount of radiation to nearby organs at risk. So I'm going to talk about the ORIGIN project, which is a European-funded project looking at developing a multi-point optical fibre sensor-based system for monitoring the radiation dose delivered to patients undergoing brachytherapy. And so predominantly this is used in prostate cancer and in gynecological cancers. And within this form of brachytherapy, there's currently no in vivo dosimetry. And so really we want to be able to monitor this dose and be able to be sure that the amount of radiation that we're delivering to the patient is optimized for their treatment and delivered according to the actual planned treatment. So the ORIGIN project looks at having a multi-point, so we have up to 16 optical fibre sensors for the point dosimetry, and then using this information we're able to develop a strategy for source localization. So in addition to having the dose, the point dose measurements for the patient, we also are able to identify exactly where the radiation sources are in real time and to be able to compare that with the treatment plan and say, do they match up? So brachytherapy is the use of internal radiation sources. So the radiation is delivered internally into the patient, unlike the other form, external beam, where it's delivered outside of the patient. And it's divided into two main areas. So we have high dose rate brachytherapy. This is where we have a high dose radiation source. So it's a single source that is temporarily inserted into the patient. And so using an afterloader, that radiation source is inserted into needles that are inserted in the patient, and it moves in steps along these needles to kind of map and paint that dose to the tumour area. And at the end of the treatment, it's removed from the patient, and so there's no long-term radiation in the patient. With low-dose rate brachytherapy, they typically insert between 60 to 80 radioactive seeds into the patient, and these are permanently left in the patient. This is primarily used for prostate cancer. HDR is used for both prostate and gynecological cancers. So the Origin project looks, as I said, to developing this multi-point optical fibre sensor system. So we insert a number of sensors. We have a bundle that goes into the Foley catheter for urethral dose monitoring. We have individual sensors that are inserted directly into, this is in the case of prostate cancer, treatment you see here. And then we have a further bundle that are inserted into the rectum to monitor the rectal wall doses. So as the radiation source is moved here in steps, the optical fibre sensors monitor that radiation dose. So in developing the sensors, we work very closely alongside the clinical teams to make sure that the sensors are being developed in a way that are easily integrated into the existing clinical workflow, because it's very important that we don't disrupt the workflow that's already established within a hospital. If we want these systems to be easily adopted into a hospital, we have to work within existing clinical workflows and not extend the time that already takes for these treatments. So as I mentioned already, we've got three main areas that we look at. So within the target area, we want to insert the optical fibre sensors into the brachytherapy needles. So we have typically between 1 and 3 dosimeters inserted for prostate treatment into the needles. And one of the main issues that we have with this is making sure that the sensors don't move during the treatment. And so we've developed these little holders here that go on the tip of the needles that lock the optical fibre sensors in place. So after you insert the sensor into the needles, you are able to then tighten them and lock them in place, and that ensures that they don't move during the treatment. For the urethral dose monitoring, we have 3 sensors. We also have electromagnetic tracking sensors so that we know where our sensors are positioned at all times. And we bundle these together with a low temperature heat shrink, and these can then be placed inside a sterile brachytherapy catheter before they're inserted into the Foley catheter, and this just ensures the sterility of the sensor system being inserted into the Foley catheter. And finally then, for the rectal dose monitoring, we have a 3D printed sheath that covers the transrectal ultrasound probe, and within this we're able to insert up to 9 optical fibre sensors and a single EM tracker. And so you can see here just a demo of one of the setups for a prostate cancer treatment with, so we use 3D printed phantoms for simulating and doing our measurements. In the white are the channels for the brachytherapy, and then we have our own sensors inserted, and additionally the sheet for the rectal dose monitoring. So these are the optical fibre sensors themselves. So what we have, it's based on radioluminescence, and so we have a scintillator material embedded at the tip of the optical fibre, and then on exposure to ionizing radiation, they give off light that's directly proportional to the amount of radiation that's received. And so by monitoring that light, we're able to monitor that accumulated dose delivered to the patient. And so the optical fibre sensors were adapted for mass manufacturability during the Origin project by a group in Brussels. And so this allows us, we were able to reduce down the size of the optical fibre sensors. So we're now using a 500 micron fibre sensor. It has, with a protective jacket of 1 millimetre, that allows us to be inserted into the brachytherapy needles. And then we have that small diameter for bundling and fitting the 3 sensors within the Foley catheter. And so here you can see that this is the scintillating tip that's attached to the tip of the optical fibre sensor. So one of the big issues then is being able to monitor this light coming off the optical fibres. And so the choice of the scintillators kind of led to the optimizing of this detector system. So with the scintillator that we've used, it has a really long decay time. And so the advantage of that is that we have this almost kind of like a multiplication factor. So for every radiation, every ray that comes down onto the optical fibre sensor, we have multiple photons coming out of the scintillator. The kind of negative of this, however, is that we need to be able to measure single photons. And so with this in mind, we use a device called silicon photomultipliers. And so we've used these, an array of up to 16 of these silicon photomultipliers that allow us, and we have choice of 2 different silicon photomultipliers depending on whether we're looking at the high dose rate or the low dose rate brachytherapy. So just looking at the high dose rate, some just initial results that we have here to show. For the high dose rate, this is a prostate treatment. And again, here you see the 3D printed phantom, and these are the white are the channels that the radiation source moves into, and the black then are the sensors that are placed within the prostate, within the urethra, and within the rectal area. So what we see here are the individual channels as the radiation source is moving in through each of these individual channels. So each of these white channels, the radiation source moves, it goes in a step, it stays in a position called dwell position for a fixed time. So if we just zoom in there a little bit more, you can see a little bit clearer how we have these steps in time. And so depending on where the sensor is placed, you get a really high measurement if it's in a channel that's close to the sensor, or it could be quite low if it's that little bit further away. So moving on then to the low dose rate brachytherapy, one of the big issues you have with monitoring low dose rate brachytherapy is that the radiation levels are really low. The radiation dose, because they're permanently inserted into the patient, is kind of delivered over about a 6-month to a year period. And that means when we measure it during the actual seed insertion, the activity of the radiation source is very low. And so the amount of light that we get out from our scintillator is therefore really low. And so to be able to achieve the sensitivity that we require in measuring these single photons coming out from the scintillator, we have to use a TE-cooled silicon photomultiplier system. And so what you can see here then is just the result from one sensor as the source is moving away from the sensor. So you have here, it's when it's right beside it, so we're at 5 millimeter from the source, and then as we move up to 30 millimeters. So 30 millimeters was the kind of the target that we wanted to be able to sense and measure at, and to achieve a sensitivity that's discernible from our noise. The 30 millimeters comes from the 30 millimeters come from basically the size of the prostate, so that it was determined that no matter where we put our sensor, at 30 millimeters, was the maximum distance within a standard prostate. So you can see here that as we move away, we're able to kind of measure that steep fall off very accurately and still have sensitivity at our 30 millimeters. And this is the first time that that a sensor has been demonstrated to measure LDR brachytherapy. So finally then, once we have the sensor readings, we combine the information with electromagnetic tracking positions, and we use that to determine the actual location of the source. So we take the sensor readings, and because there's an isotropic function of the radiation source, we use a machine learning approach known as Bayesian inference to determine the most likely position of the source relative to the sensor. And then using that information, combining it with the actual position of the sensor from the electromagnetic tracker, we use just a standard trilateration approach to be able to pinpoint where the actual sensor, where the source is. And then finally, once we have that information of the source's location, we're then able to build up an overall dose map. So in addition to the point dose measurements that we receive, we then have a dose map with these isodose contours of the overall dose being delivered to both the tumour and to nearby organs at risk. And this just shows some of the preliminary results for that. So finally then, within the Origin project, we've been able to demonstrate the use of optical fibre sensors for brachytherapy dosimetry. We're able to get the sensitivity required for the clinical relevant distance for both HDR and LDR with good precision and good spatial resolution. And then combining that with the machine learning algorithms, we're able to use to determine the actual source location of these radiation sources during the treatment. So just to thank all of the partners, both the universities and the clinical partners, and obviously the funding agencies. So thank you all very much.
Can I ask a question? It might be a bit, it's completely not my area, it's really interesting. What increase in sensitivity do you think that your system will provide? So presumably you're going to be able to provide better dosing to patients and improve dosing regimens or...
Yeah, so that's really kind of the long-term aim from the clinical point of view, is that if they have the information of the radiation doses, they can take kind of larger risks, so they can give higher doses at an instant. So at the moment for HDR, for example, patients would typically come in 3 times for their treatment. And so ultimately, they'd like to reduce that down to 1-2 times, reduce the number of times that patients have to come in for their treatment. And so they call it dose escalation, that they give a higher dose. But when they're not 100% sure of what the radiation doses are, particularly to the organs at risk, they tend to kind of err on the side of caution. And often you get that underdosing because they just don't want to risk.
So you kind of think of personalised medicine as kind of with drugs, this is personalised medicine.
Exactly.
With kind of physical side of the dosing. That's
Yeah,
brilliant.
that's it.
Oh.
Yeah, and just a quick short question onto that. This is fantastic work and so important for patients. We sometimes end up avoiding radiotherapy, particularly to the bowel, say for my lymphoma patients, because of the toxicity. Yeah. Is this being looked at in other areas other than prostate? Or?
So at the moment we're working with the prostate and the gynae consultants on it. We haven't looked beyond that because really just brachytherapy is only being used for those patients. But I think if they can start to deliver brachytherapy with greater confidence, then it definitely, will start to roll out kind of other areas. Yeah.
Definitely something more for some panel talks later.
Thank you so much.
Thanks. I'd just like to thank all the speakers, a wonderfully diverse sort of range of things that are going on. So now we have time for a short panel discussion. So we'll get all the speakers to come back up if they'd like, and some other.
Paul just messaged me that he's waiting in the lobby. I think there's a few of them with non-email addresses. Do they need to be let into the meeting online?
Also the online. So we can get the speakers down anyway. If there's any other questions. I'm going to preempt Kieran here and between you and I. I'm just curious about the endometriosis. Why don't those cells become tumorigenic if they have so many characteristics across the cancer hallmark? Why do you not commonly see endometriosis converting to tumorigenic?
I couldn't tell you, James. I couldn't honestly. That's only that paper only came out last year, and it kind of chipped through off everyone's thought processes where people started to think, was there a need to kind of recharacterize this condition? It wasn't falling into that benign, classic trap. So I think those groups will be working on that and seeing, but there still is no evidence of transformation.
Yeah, it's just interesting because one of the main things I teach the students in fourth year is about NF-kappa B, that it's central to so many of those hallmarks. That's really interesting to see.
Yeah.
Kieran is flying.
I was just interested in do you think with finer sensor and localized measurement of dosage that you could inform further response and that you end up with tissue-specific dosage regimes that start to evolve because you'll know what is an effective dose or you'll know what would be required in that tissue type?
Exactly, yeah. So they are looking to kind of use that information. Once they have the information on the dosimetry side of it, then they can combine it with their patient outcomes-based research and combine that and say, where is the link, between the outcomes and the in vivo dosimetry. So currently they're just using kind of computerized models to say what they expect the dose to be, but they don't actually know what the dose is. So if they can combine that, we're also looking at sensors that would, say, combine with the radiation sensors that would look at the actual tumour microenvironment. So, and again, combining that information to look at where the radiation,
Intersect is that?
Exactly. Yeah.
Máire?
Hi there. So my name is Máire Lavelle. I'm a consultant pathologist in UHL. Now, I just have, it's not really a question, more of a comment for Jason about endometriosis. So, I think you just hit a nerve with something that affects so many women, and it's so common. And I would see endometriosis in my work, and it often imitates tumour masses. And I have seen surgeries, huge surgeries done, and when we put it under the microscope, it's actually endometriosis. And even microscopically, it is a pitfall for pathologists to make sure that we don't accidentally diagnose it as adenocarcinoma, because it can look like tumour cells under the microscope. But I was just thinking back to my kind of embryology days. I think in terms of imitating tumour, it just reminds me of my old anatomy professor kind of saying that the gynecological tract, a lot of the gynecological tract imitates tumours. So when the fertilized ovum goes into the uterus, it invades into the lining of the uterus, and we don't really understand how it knows to stop invading. I quite liked his comment was that, and then it keeps invading and acts like cancer for another 18 to 21 years at least. But I suppose it's just a comment is that I'd wonder in not just endometriosis, but maybe in normal endometrium or in other areas in the gynae tract, would you see similar profile in terms of of that? I know it's probably not something you can answer, but I suppose it's something
I can't
to
answer that, but
think
like,
of.
obviously, if we get where I want to go in the next 2 years, where we're actually analysing tissue from patients, like with every endometriosis sample, we'll be looking at utopic tissue as well, and you know, once we start to pinpoint exactly what immune cells are there, what phenotype, hopefully, you know, the curtain will come down a bit more. Actually, what's driving this condition?
Well, if you need a pathologist to look at any slides for me, you know where.
I need all the help.
Can I ask a quick question, which is, sorry, I'm kind of coming on the side here. Like with regard to the brachytherapy sensors that are in situ, what's the potential in the future for kind of real-time monitoring of other measures in the tissue that might indicate successive treatment?
Yes, so we are looking at tumour hypoxia, for example, and looking to see how that, because tumour hypoxia is one of the big issues around kind of the failure of radiotherapy. If a tumour is particularly hypoxic, it tends to be more resistant to radiation. So that's one of the areas that we're looking at in measuring it. And so the optical fibres that we're using have, because of they're so small, you know, they can be kind of minimally invasive in terms of getting them into areas that are, you know, that a lot of the existing sensors are just too big. You know, we're able to fit our sensors within existing brachytherapy needles, within Foley catheters, and it just allows for that kind of use of getting sensors. So while we have currently just the radiation sensors, there's nothing to stop us, and we do have a few looking at the more sensors related to the tumour microenvironment and trying to look at that. And I think that's a whole big new area that I think has huge potential.
So I was just wondering if you both are working together, you know, for grant applications, infertility, etc., or is this an opportunity?
No, I only joined last year, so I'm still trying to find my place and see who's doing what. So that's why.
Well, you know now, so.
I'm sure we run the market really, so
All right, I think we're kind of done for this session. I just want to thank everyone and the speakers for showing the wonderfully diverse range of research and techniques that's going on at the Cancer Centre. Just in this first session, you can see we've got a range of different cancers, a range of different techniques, people at all different stages. So I think it's really good to highlight during Research Week just some of the work that's going on, and hopefully you can come back at 4 for the second session where you'll get an equally diverse, or 5 to 4, sorry, 5 to 4. So quick coffee to hear about some of the other really interesting work that's going on at the Cancer Centre. Thank you very much.
Okay, we'll get started with the second part of our session. And we've just got a little surprise video that we felt it was very important to show today. So the audience for this video is really the patient and the public. But we want to outline that we have a patient research group that we've brought together. Myself and Miriam McCarthy, who's sitting in the back there, kicked this off about 2.5 years ago now, is it? Or 2 years ago? 2 years ago. And we have patients from the Midwest region coming together with some of our academic and research nurses to be available to help us with our research from, you know, the get-go. So we're trying to expand our group so that we have more patients involved. And Dr. Elizabeth Ryan applied for a PPI Ignite grant and was able to put this nice video together highlighting what PPI is for the public and patients, because we're going to go on a recruitment campaign now to increase the number of patients who are participating in our research. So I'm just going to play just a minute and a half or so of the video, and then we'll move into the next session. Sorry. Sorry about this. So look, it's just really important that us as researchers understand the importance of PPI and the relevance, and that it's not just a tick box exercise. So we do want any of our researchers who are participating in working with our patient group to be informed about this. And PPI Ignite have, what did I do? Here we go. PPI Ignite have training for researchers and we're very keen that you participate in this training. So if I press play here, I'm not going to ruin it again.
Or by patients or members of the public rather than on, about or from them. PPI means involving people as equal partners at all stages of cancer research from start to finish. Yes. Your lived experience as a patient or carer is the most important qualification. No prior PPI or research experience is needed. Also, training is available should you wish to learn more about the research process. You will work alongside another researcher or mentor on the team to help you get started.
Patient researchers can volunteer for the projects that are of most interest to them. Some stages of a research project might be busier than others. For example, at the design stage, when there may be several documents to review. However, generally, you would meet with the research team once a month or so. Meetings can be arranged online or in person and at a time that is convenient for you. Knowing that my contribution will influence and shape research that will lead to better treatments and outcomes for all cancer patients is fulfilling. I take pride in doing this valuable work that will advance cancer research and treatment and find it rewarding. If you feel that you would like to contribute to cancer research in the Midwest, I encourage you to get in touch and find out more about joining the Midwest Cancer PPI panel. You can find out more information by contacting Liz O'Leary.
The most important reason to do research is to improve the quality of care that patients receive. The better we understand cancer, the more progress we can make towards developing new treatments and better diagnostics.
Okay, we all know the answer to that question, I suppose. So I hope you got a nice flavour there of what our group are doing. And yeah, we will feedback more at our next meeting, I think, which is in June, on that. Thank you very much. Okay, so we'll start our next session. Tumours in context. Unfortunately, our chair has been could not make it this afternoon at short notice, so I'm going to just take over there and introduce Miss Christina Fleming, colorectal surgeon from UHL. We're delighted to have you here today to speak to us about colorectal cancer surgery.
Great, thank you, Ruth, for the introduction. So, as Ruth outlined, I'm a consultant colorectal surgeon, so my talk is going to be very clinical and a little bit different maybe to the previous talks. And I'm going to really focus on some of the complexities of delivering research in a clinical context when it comes to complex interventional therapies like an operation, particularly looking at the application of robotic surgery with rectal cancer and then advanced rectal cancer where we might remove more than one organ. So here are just some financial disclosures relating to robotic surgery that I would have. So I'll talk a little bit about my own background, a little bit about technology-enhanced surgery, what this is, how it's evolving, the ideal framework, which essentially is the roadmap that we use to assess the efficacy, safety and effectiveness of surgical interventions. And then finally looking at kind of robotic pelvic exenteration for advanced rectal cancer, really as the kind of newest thing that we're, I suppose, doing to push the boundaries for improving resectability and reconstruction for pelvic cancers. So I'm a UCC graduate, but a proud Kerry woman. And following my graduation in 2011, I did what would be, I suppose, described as a surgical tour of Ireland, where I moved around for many years, learning to perform different types of surgeries across general surgery, but always with a specific interest in colorectal cancer. And then following my training, I moved to Bordeaux. I learned to speak French, and I completed the European Society of Coloproctology Robotic Fellowship. And whilst most people who came to visit came to see the beautiful historic architecture and the vineyards and taste the lovely wine, I spent most of my time there looking at these kind of scenes. So really learning everything there was to do with resecting rectal cancer, from minimal resections, which would preserve the actual rectum, right up to pelvic exenteration, where we were removing part of the sacrum, reconstructing iliac vessels, and everything in between. So during this time, I always had somewhat of an academic focus, and I took time out of my surgical training to complete a PhD, which was a collaboration between the BIH at the Max Delbruck Molecular Medicine Centre in Berlin and UCC, and this was really focused on metabolomics and perioperative cell-free DNA and looking at how prognostic that could be in colon cancer. I've also chaired the Irish Surgical Research Collaborative, which is a national group looking at trying to bring all of us together across the surgical community to produce high impact, clinically relevant research. And also I have done a lot of research in surgical training as well during my time with ASSET. So I feel like I am uniquely placed to describe where do surgeons add the most value when it comes to research. Now, obviously I feel like we add value everywhere, but I think really it's in surgical intervention or surgical innovation and introducing new surgical techniques where nobody else can really do that unless we demonstrate leadership in that realm. So if we go back to the origins of the word surgery, it really means doing work with our hands. And whilst we still use our hands to perform operations, the patient interface that we face every day is very different, and it's very technology enhanced. So this is how I would routinely perform a rectal cancer operation, sitting at the robotic console with this big device attached to the patient. And you can see here I'm doing some dissection in the posterior mesorectal plane, and you can see all of the nerves falling inferiorly. So this is how we're performing surgery now. But how did we get to that point? And how did we use research to get to that point? So I feel like surgery is in this, evolution from 1.0 of big open incisions and, lots of blood loss and lots of morbidity. And we're moving more and more towards precision. So all of these individual types of operations do still have a place, but I think we're moving far more to the right of the screen in more frequency. If we look at this first video, this is a laparoscopic resection where we're dissecting the rectum away from the vagina anteriorly. And as you can see, you have a decent view, but you don't have an excellent view. But if we move to the next video, where we've introduced robotic surgery, 3D visualization, better instrumentations, I think most people can make out exactly where the vagina is anteriorly and exactly where the rectum is posteriorly. And that precision equates to better outcomes for patients. And then how do we make this digital? Well, we use adjuncts and add-ons. This is ICG. We administer this to patients so that we can know exactly how well the rectum or the bowel is perfused. And with that, we can decide where is the best place to perform our anastomosis, reduce anastomotic leak, and optimize patient cancer and clinical outcomes. So a lot of the talk I'm going to talk about robotics, and why talk about robotics, and why do we use robotics in the setting of rectal cancer? Well, all of these publications have come from our colorectal unit in UHL, and essentially what we have demonstrated in our own patients is that applying robotic surgery in colorectal cancer reduces complications, surgical site infections, hospital length of stay, conversion rates to open, particularly in more complex cases. It increases lymph node yield, and also is associated with improved functional outcomes, particularly with urogenital function following rectal cancer resection. But I guess when robotics came on the market, not everybody was a fan. And there is a legacy of, bad news stories of when surgical innovation is introduced in the wrong way, and it can lead to really devastating outcomes for patients. And obviously in the bottom right, we would all be very familiar, I guess, with the Temple Street story in the past year with regards to spinal rods being inserted, maybe not in the most quality assured way and not following the right framework for their introduction. But randomised controlled trials to, I guess, assess safety and efficacy are very challenging in surgery. This is the seminal robotic rectal cancer study that we have. It was a negative trial. It added nothing to the literature and didn't help us in any way. And why is that? Well, surgery is a complex interventional therapy. There is the operation itself, but there's also patient anatomical factors. There's the surgeon themselves, their factors with regards to their learning curve. What do I mean by that? Well, have you performed enough of these operations to be defined as expert? And obviously technical skill can be different amongst different people. The experience of the operative team, how do you control for that within a trial? And how do you control for the large perioperative system within our hospital? Well, all of those things are actually very challenging. And that's why we're kind of moving back now in robotic surgery by realising we weren't probably ready for this trial when we did it, to performing more kind of cohort-based study in expert centres where a lot of these factors are far more homogenised. And that brings us to the ideal framework. And this is just a roadmap really that we use for studying complex interventional therapy and identifying, well, where is this, innovation or surgical technique at in its development, and what is the best way to design a study to bring it forward even further. And this is the kind of flowchart that we use. And, if we look at the study types and the methodology in the different stages on the right-hand side, they're all quite similar to what we might use in a drug trial, but they're just a little bit more nuanced. And the descriptions for what the different criteria are at each stage are very much focused on safety and efficacy, because a lot of the time these are being implemented in an actual clinical setting as well. I might just skip through that. So where are we with robotics now and robotic surgery, particularly for pelvic cancers, and how do we kind of get to that stage? So if we look across the first line, so robotic surgery, pretty much there's over 20 years experience of performing robotic surgery for endometrial cancers. It is the standard of care for prostate cancer. Over 85% of prostate cancers prostatectomies performed for cancer in the UK are performed robotically, and I think it is now the emerging standard of care for rectal cancer as well. But when you put all of these procedures together in one operation, we call it a pelvic exenteration. And I'll get onto what that is in the next few slides. But if we apply robotic surgery to the combination of all of these three techniques, we actually have to bring our framework back to stage one. So when we perform surgery for rectal cancer, we essentially remove the rectum and the mesorectal fat with all of the lymph nodes that are associated with it. And we know from seminal work by Bill Heald and his group in the UK that this offers patients a fantastic 5-year survival rate. But what do we do with this kind of cancer that involves not just the rectum, but also the bladder and the uterus? Well, this is where pelvic exenteration comes in. It means removing en bloc at least 2 pelvic organs with or without diversion. So if you're going to remove the bladder, how are you going to get urine out after that? You need to think of how you're going to reconstruct all of these systems that you're resecting. The aim is to get an R0 resection, so that's a clear margin with at least 1 millimetre. And we're pushing more now towards reconstructive methods as well, and I'll describe them a little bit. So this is what I mean when we talk about pelvic exenteration. We tend to describe it as, it can be performed for primary or recurrent cancer. An anterior pelvic exenteration would involve removing, let's say, the bladder and the uterus, or just a radical cystectomy in a male. Posterior, generally the rectum and the uterus, or the rectum and the prostate. And a total pelvic exenteration would involve removing all of the pelvic organs. And obviously, this would be considered a soft tissue pelvic exenteration, but there can be add-ons with vasculature and bony resections as well, depending on tumour involvement. And once we resect everything, we have to think about putting it back together. And I would think that reconstruction in this setting really has only I think, become, more commonplace in the past 5 years. So, prior to that, if you had a pelvic exenteration, you had bags everywhere. You had a stoma bag for your bowel content, a bag for your urine. And obviously, if you had a vaginectomy, everything was just sewn up, and that was it. And you were just grateful that you had your cancer resected and you moved on. But we're moving now far more towards a survivorship model, where, we can cure the cancer by removing it and getting a clear margin, but that's not at a cost of everything for the patient. So this data is a little bit old, but it's been pretty well replicated in global data as well, and it's from the National Cancer Registry, looking at over 40,000 patients over close to a 20-year period. And what we're seeing more and more are younger patients under the age of 50 presenting with these really advanced, aggressive rectal cancers. And we know from multiple data sources that by 2030, over 50% of rectal cancer will be diagnosed in patients under 50. So we need to get better at resecting these cancers and reconstructing systems after that to try and optimize survivorship for patients. And there is huge oncological benefits to performing a pelvic exenteration and getting a clear margin. So this is from the Pelvex Collaborative, led by Michael Kelly, who's a colorectal surgeon in St. James's, who I trained with. He's a very good friend and colleague. And they've demonstrated that if you can get an R0 resection, it's probably achievable in about 85% of people, and 3/4 of them will be alive at 3 years following their surgery with an acceptable quality of life. So why perform robotics in this setting? Well, we know with minimally invasive surgery, it offers reduced blood loss, reduced morbidity, and reduced hospital length of stay, and gets patients back to work quicker, which obviously for younger patients becomes very important. But performing laparoscopy in the pelvis is really challenging with the bony limitations that are there. And the management of bleeding is very complex because you have less arms to work with. So robotics kind of overcomes all of these laparoscopic limitations whilst also offering minimally invasive surgery benefits. But if we look at the data, there's only been 4 studies that have actually looked at robotic exenteration in this setting, all kind of sitting between the stage 1 to 2A of the ideal framework. And when I was in fellowship, this was a central part to the academic theme of my fellowship, and actually kind of like developing and evaluating and improving our robotic technique for performing these kind of surgeries. And we had a phenomenal reduction in hospital length of stay, blood loss, overall morbidity, and particularly severe complications. And currently, we've moved into the stage 2B phase. This is being led by a group in the Peter Mac. This is ropes. We're currently at the Delphi stage of kind of informing all 3 work streams. And hopefully over the next 5 years, this is really going to inform how we apply robotic surgery to pelvic exenteration. And then just to finish up, I'd just like to really demonstrate the real-world patient impact of this kind of approach and how we can, apply more minimally invasive surgery to advanced pelvic cancers. This is a 44-year-old woman with recurrent low rectal cancer. She was first treated in 2019, who presented with a malignant rectovaginal fistula and had some margins kind of abutting but not involving her right ureter and right-sided levator ani. So in 2010, this would be deemed unresectable, sent for palliative chemotherapy, probably die within 6 months, a horrible death from local invasion of all of her nerves, and sometimes these even grow on to the anal margin. By 2015, it would have been deemed resectable, but there would have been no option or minimal option for reconstruction. But now we're in an era where we can resect these kind of big tumours using minimally invasive surgery with reconstructive options. And I might just show a very short video as to how we do that. So here we're using the robotic system, where we're actually going to perform a rectal resection on block with a total abdominal hysterectomy and bilateral salpingo-oophorectomy. As you can see on top of the video that's playing at the moment, we performed all of the surgery through these ports, so there was no big incision down through the abdomen and the morbidity that that's associated with. We're dividing the IMA now, so we can remove all of the lymph node package and continuing our dissection posteriorly along the mesorectal plane. So as you can see, this is very precise surgery, excellent visualization from the robotic platform, and we can see all of her nerve fibres sitting posteriorly. We're now just going to perform ureterolysis along the right ureter, as we know that there's a really close margin. This is actually all really challenging to perform laparoscopically, and really where the robot kind of comes into its own. And once we get to the anterior dissection, we'll move over to the other video. So once we've resected the tumour, we need to think, well, how are we going to put everything back together? And she really didn't want to have a stoma. So we decided to do what we call a delayed coloanal anastomosis. And the cartoon kind of picture is to kind of depict what we do in that setting. So we essentially bring the colon out through the anus. We leave it there for 4 days. It turns black. We cut it off and we suture it onto the anal canal. But what does that allow us to do? Well, it allows us an opportunity in the immediate and post-operative period, as we're going to see from the video on the right, to synchronously perform a vaginal reconstruction. And we're not going to have both of those anastomoses healing themselves at the same time, so therefore reducing the rate of them fistulating or causing complexity. So here we've just essentially pedicularized a little section of her small bowel, which we're going to fully open up along the anti-mesenteric border, and we're going to reconstruct a vagina with this. The video's only about another 30 seconds, so I might just leave it, because I think it's a really good example of how we can, use patients' priorities in their care to inform what we do from a surgical perspective. So we're essentially, we've everted the actual small bowel conduit. So this is what the neovagina essentially will look like. And following we just put some swabs in so it can maintain its structure, and we place it back into the abdomen. And then we essentially anastomose it down to the enteritis. Now this is just a structural reconstruction, it doesn't really have a function, but it's well vascularized. And again, I think we use ICG in this video to confirm the vascularity of both, the vaginal reconstruction. You can see the colon sitting out inferiorly as well. So I guess in summary, robotic colorectal surgery is allowing us to really enhance clinical cancer and functional outcomes, and probably push the boundary with resectability as well. Researching complex interventional therapy is complex, and we have to accept that. And I think the ideal framework is a really good way of helping us navigate those nuances to ensure that we can produce high quality research from these techniques as well. So thank you.
So impressive, Christina. Thank you very much. Is there any one pressing question from the audience? James?
It sounds really great, but I'm just wondering what sort of training interventions do you need to have at a medical school level? What sort of training interventions do you need to bring into it? And does this mean that in 10 years students won't be taught kind of open surgery, that the primary way of treating patients will be through robotics?
So I think laparoscopy will probably become obsolete in the next 10 years, and it will be open or robotic. You'll always need to be able to perform open surgery, because when something goes wrong, that's how you save someone's life. So from even a trauma point of view, you'll always have opportunities to learn open surgery. But robotics are probably going to, I think, surpass laparoscopy. And I mean, I'm involved in multiple training groups with regards to robotic surgery, specifically for medical students. Last Saturday, we'd RCSI SPRs down in our theatre to show them how to do right hemicolectomies, using the robotic system. And, everyone is recognising now this is really important, and there's a lot of investment of resources and time going into it.
And can I just add to that, how well placed are we in Limerick? Because this sounds like something that we can really expand on.
Yeah, so the department here was the first department in the country to introduce a robotic colorectal surgery programme. I was trained in that programme before leaving and coming back as a consultant. So I think that in itself actually demonstrates where we're sitting and leading on a national level. And I think, we are one of the 8 NCCP approved rectal cancer centres as well. So I think there's huge opportunity for us to continue pushing on and pushing the boundaries in this realm. So, yeah.
Okay, fantastic. Thanks a million, Christina. And now we'll move on to Kieran McGourty, who's going to talk to us about cancer stem cells in the tumour microenvironment. Thank you, Kieran.
Thanks very much. I just want to start with actually thanking the organisers, because I don't know how everyone else feels, but I think it's been a hugely, hugely positive day, and really highlighting the links between the hospital and what we're trying to do here at UL, and how important those links are. So I just want to thank the organisers and for the opportunity to speak. Now, by rights, I'm speaking here, but this is all work that's associated with one of my PhD students, Seamus Hickey, who's also co-supervised by Pat Kiley. in the audience, and Siobhan Cashman. And Siobhan is working in BD Biosciences that are also co-located here on campus. And again, I think that highlights as well another very important arm that I think we're excellent at here in Limerick, which is involving industrial partners. So this is a nice illustration of a industrial academic partnership and how it can yield some very interesting data. So Seamus would undoubtedly do a better job of this than I, but I'll try as best I can. And my major contribution to this work is last night I came up with a description of what we're describing here, and I'm going to call it Sleepy Hollow. So these are regions within the tumour microenvironment that I think allow stem cells to enter a dormant state. Okay, so tissue is highly organized. And it's continually being renewed, and it can respond to major trauma and insult. And a lot of that capacity is associated with a population of cells known as stem cells that reside in regions of very defined architecture within the tissue. So this is the pointer there. So each tissue is different, but they share some organizational commonalities. So usually there's a region in which the stem cells reside. They're juxtaposed right next to them by a proliferating population that then goes on to differentiate to become, in the epithelial tissue, the differentiated cell types that make up that tissue. Tumours are also organized, but not consistent between tumours. So there are microdomains within tumours, there are regions of recognizable cellular structure within tumours, but between tumours, it has lost this repeatable organization that we see in healthy tissue. In tumours, there's also been reported that there's a population of these stem cells, these cancer stem cells, that are very important for the disease progression, and are also have been reported to reside in specific regions within the tumour. How those forms, those niche environments form, what triggers that stem cell to either reside and stay dormant, or to enter into a proliferative state here in the healthy tissue or in the tumour, an expanding cancer proliferating cell, is not known. The triggers that induce that dormancy or entry into the cell cycle are not well described. Okay, so Seamus had this understanding that cancer stem cells were important, and that they can reside in these microdomains, and that a certain population of cancer stem cells have been reported to enter dormancy, just like healthy tissue stem cells. He also was very interested in these because they're extremely rare, making them difficult to characterize, which is why there's such a paucity in the literature around understanding how they behave. They're also therapeutically very important because they tend to be resistant to many chemotherapies by virtue of the fact that a large population of them are not replicating, and they are believed to be one of the reservoirs that allows for re-establishment of cancer after therapy. Okay, so we were approaching this in our lab. We do both practical lab work, and we're also moving increasingly to looking at data sets, particularly single-cell data sets, single-cell sequencing data sets, and multi-omics approaches, based on the cooperation of people and the many published data sets that are out there that allow for mining of this sort of information from studies that have already been undertaken. So Seamus assembled this large cohort of 82 patients across different regions that were colorectal cancer patients, and he wanted to search and sift within this data set for a subpopulation of cancer stem cells. So this data set he took forward, and he started to look at the architecture of the healthy tissue and what is known and what's described about them, and what is known and described about the cancer stem cells in cancer tissue. And one of the key components that marks cancer stem cells in both cancer and healthy tissue is this protein marker known as LGR5, and that these tissues, these cells really are a divergent path away from what would normally be the normal homeostatic dormancy and replication that occurs in healthy tissue epithelium. And this recently has been very well mapped as the signatures of the different cells that comprise this healthy epithelium. So Seamus was able to assemble his large metadata analysis set. He integrated those sets into a single cell data set that comprises cancer stem cells, and to verify that he was actually looking at cancer stem cells, he selected those cells that are LGL5 positive and selected those cells that would be to the other end of the more differentiated spectrum of this trajectory, which would be CCAM7 positive cells. And he was seeing that in his data sets, he was getting clear signature and separation between LGR5 cancer stem cells and those CCAM7 positive cells that were also within the TME. We verified these with secondary markers of stemness and those secondary markers of differentiation. And again, we exploited the current literature to give this an axis, a crypt axis score, or a score of stemness. And we could see that the population that he'd enriched for were indeed heavily associated with stem cell markers. So we went on to try and explore these, and he wanted to cluster them. And the first question he had were they all the same? And what we saw was that they clustered differently in many different subclusters, immediately indicating to us that these are transcriptionally different populations of cells, though they're all falling under the marker of a cancer stem cell. And this is novel. It was known that there was this dormancy and proliferation cycle, but it had mainly only been described in one canonical marker of P27, and a single ECM component, collagen 17, had been implicated in that control. This large degree of heterogeneity that we're observing here wasn't seen before. He went on to look at this in healthy tissue, and he saw that he was getting a similar degree of heterogeneity in healthy tissue, indicating that this may be a normal process of the cancer stem cell behavior. And he went on to see that were these behaviors similar in both healthy and in disease case, and he saw that they broadly clustered together against canonical markers of dormancy, CDKN1B, which is P27, and two new ones, P57 and P21, that have been reported in the literature are important for maintenance of this dormancy program. And they are associated with this upper cluster here and this lower cluster here, with P27 being in the center. And these were replicated in both healthy and disease cases. And then there's a proliferating cluster and going on to a cluster expressing markers of differentiation, indicating that this may be a dormancy axis that's then emerging into a proliferation and going on to differentiation within the stem cell compartment. We went on to start to characterize these different clusters, and we could see that quiescent P57, quiescent P27, or quiescent P21 all displayed different hallmarks of deep dormant quiescent behaviors, such as elevated Wnt, elevated P53, and downregulation of MAP kinase signaling. And this was also evident in the healthy tissue, again showing the commonalities between this process, which may not be actually oncogenic, may be just a product of the environment that that cancer stem cell finds itself in, that it activates a healthy dormancy program. So the question was, we really centered this on whether the microenvironment would also be specifically regulated to these clusters, and Seamus was able to see from the differentially expressed genes that he was getting unique signatures of extracellular matrix genes that were associated with these clusters that were specific to these clusters, and they were in large, common between both tumour and in healthy tissue, though there were some differences that I don't have time to go into today between the extracellular matrix environment in healthy tissue compared to tumour. So then Seamus came up with this really fantastic challenge of a hypothesis experiment, in which he said, okay, if these cells are really expressing a unique ECM environment, what happens if I only cluster those same cells on ECM? Do they cluster separately first, and then do those behavior, which are other genes that are not associated with the ECM, also go with that clustering? And what he saw was that indeed he was able to get this separation, this quiescent population again, displaying key signatures that we had P21, P27, and P57 that were clustering separately then from the clustering separately when we only looked at the ECM, and they co-clustered with them the same behaviors that we were seeing. previously when we clustered on the whole transcriptome. We took those populations and we did an agreement matrix, and we could see that for everything but the proliferating population, there seems to be a large degree of agreement that the matrix is governing that quiescent program. And in both healthy and in disease case, we're getting a high degree of correlation between those same cells clustering, whether we look at their matrix expression or we actually look at the behavior markers that are in that environment. We went on to describe that and develop what we think is an extracellular matrix signature of this behavior, and we called it P21, U cell, P27, P57, proliferative one, a committed one in both healthy and disease cases again. And we projected that back onto our whole cell data, and we could see that those ECMs were particularly enriched in each of the clusters that were canonical of that behavior that we saw previously. Independently, because we were worried about our data set and whether there could be an artifact of integration, we adopted a secondary approach in parallel known as non-zero matrix factorization, in which we were able to take those 82 patients and we were able to treat them in isolation, so you don't have to integrate your data at all. You can take those 82 patients, we can assess their transcriptomic performing internal to each individual patient, and then we can do that across all 82 cohorts under the understanding that true signal will be reinforced, whereas any stochastic noise, because these are large data sets, would drop off. And in this, we were able to develop these clustered meta-programs, so they're gene sets again, that are associated with specific clusters of cells, and we call these meta-programs 1 through 3. There's 8 of them there that we described. And what was nice was, so this is our P57 cluster, for example, here, and this is our P21 cluster. We were getting specific meta-programs that were lighting up associated with the behaviors that we were seeing clustering on the whole cell. And this, again, is a completely independent, non-integrated version, but within the same data. We went on to project that back onto our integrated data set, and we were able to see that those meta-clusters centered around the same regions of dormancy that we predicted would be the clusters associated in the whole cell clustering. And then to bring a long story back to another great test that Seamus came up with, he was saying, okay, now we have a signature on the ECM, we have a signature on these meta-programs. Could we use that to project and identify regions of dormancy within a naive data set? This is a data set we haven't seen, and we adopted the same approach. We took a spatial transcriptomics approach from a Visium data set in which we subset out, again using the same markers, the stem cells within that colorectal cancer tumor sample, and then we projected, or we scored those cells against our signatures. So we took the ECM signature and we took the metaprogram signature, and then we were able to see that it really nicely described microdomains within the tumor microenvironment that clustered together when we clustered on these signature scores, that clustered together into discrete islands of proliferation or dormancy dependent on the signature that we were looking at. We overlaid that again. We have an independent interrogation of these meta-programs, and again, these meta-programs, particularly meta-program 3, for example, here, and this island of dormancy here that's associated with P57 and P21 is also heavily enriched with this meta-program 3 in quite a discrete fashion. Okay, so ultimately we brought this forward and we did a correlation matrix, and we saw that there was a high degree of correlation between the signature, between the meta-program and the NMF, which makes us prompts us to believe that we can now describe these dormant niche environments within the tumor, and that is going to be somewhat cell agnostic. So whereas we can describe here stem cells, we can also look now at immune cells in that environment, which there's reported instance in the literature as that environment being immunosuppressive, which is very important, for example, when you think about immunotherapies and whether they will be applicable in different forms of cancer. I won't go through the summary, I'm just going to go through the thanks, which is more important. and that's Seamus. He's fantastic. He would have been here today, but he asked for a day off very rarely to go to his brother's wedding. I reluctantly agreed. Actually, I wasn't asked. And then these are two other collaborators whose lab I'm in. This is Dr. Santiago Carmona, and this is a senior bioinformatician research scientist. I'm on sabbatical in the Eoghan Cancer Centre in Unil over in Switzerland, and they've helped a lot with the signature scoring and the projection analysis on this. So, and also as a really nice collaboration and the speaking to the ability of tools, Seamus and others in my group are plugged into their Slack channels, so as they develop, there's continual communication back and forth, and I hope that becomes a real access for Limerick going forward.
Any questions for Kieran? Or we can keep it till the discussion, maybe, and we'll move on. Yeah, okay, fabulous. So next, I'd like to invite up Ciara Leahy to talk to us about primary Epstein-Barr virus and EBV-associated malignancies.
Thank you very much. So it's been a bit of a whirlwind week for me. I literally just defended my PhD thesis last Thursday. So thank you very much. Unfortunately, the amount of time prepping for this presentation and the amount of time celebrating weren't exactly well balanced. But hopefully I will be able to give you a taste of what I did during my thesis and what I'm going to continue to do over the next few months in the lab of Professor Paul Murray, where a lot of our research is focused on the Epstein-Barr virus. And my thesis specifically looked at the spatial immune landscape of EBV-associated diseases, including benign and malignant conditions. So as a brief background for people who don't know, the Epstein-Barr virus is a virus which infects over 90% of the world's population, most of whom are completely healthy and never even know they're infected. But in certain circumstances, EBV can cause diseases, some which are benign, you might have heard of like glandular fever, and some which are malignant. But the cell type that EBV preferentially infects is B cells. So a lot of the malignancies associated with EBV are B cell lymphomas. But the virus can also infect other cell types like epithelial cells and NKT cells, meaning that there are also EBV-driven malignancies in cancers with these cell types of origin. Most of EBV's oncogenic capabilities and pathogenesis are attributed to a protein called the latent membrane protein 1. And this is the protein which a lot of studies have been done on in vitro in EBV-associated malignancies. So as a general overview of what my thesis was, which obviously I don't have time to go into everything today, I looked at using spatial biology techniques to explore the immune landscape of EBV-associated diseases and how EBV and the immune system interact. And I'll make a little confession that when I read the email asking me to speak today, I read that the talk was meant to be on the tissue microenvironment and not the tumour microenvironment. So out of the three diseases which I looked at, only one of them was not it was not a tumour, it was rather benign malignancy, and that is the one that unfortunately I prepared my slides on today. But what I did in infectious mononucleosis is very similar to what I did. They're the same techniques, the same process, and I'd be happy to talk to anyone about my work that I did on NPC and ENKTL as well. And we've also looked at other diseases in the lab, including DLBCL and Hodgkin lymphoma. So as I said, infectious mononucleosis is a benign condition which is caused by the Epstein-Barr virus, and it happens when a person is first infected with EBV, which we call primary infection. And when I went to the literature to study IM as part of my thesis, it was kind of forgotten about. It was studied initially up until about the 1990s, and then people kind of stopped caring about it. And I can only assume that's because it's a self-limiting condition, it's resolved, and people became much more interested in EBV-driven cancers. But I thought this was kind of a unique opportunity because EBV only expresses about 10 proteins in all of the malignancies associated with both benign and malignant. And I think it's very interesting to see the difference between what happens in benign conditions and what happens in malignant conditions, and whether we can use those differences to learn something from what's happening, and how the immune system kind of responds differently or similarly in these conditions. And furthermore, infectious mononucleosis is also a risk factor for developing EBV-driven cancers later in life, including Hodgkin lymphoma. So again, I think it's of importance to study IM and see if there's anything that can tell us about why it might be a risk factor for other cancers later in life. So in terms of the methods that I used in my thesis, the main method that I used was the phenocycler fusion, which is also called codex chemistry, or used to be called codex chemistry. So what it essentially is, it's a microfluidic system called the phenocycler, which is attached to a microscope called the fusion. And what it does is it allows us to look at multiple proteins at the same time in tissue. So the process by which it works is that you get all your antibodies to the markers you want to look at, and you conjugate them to DNA barcodes. You then stain all your antibodies onto the tissue and fix them in place. And then in cycles of 3, you image those antibodies and you repeat that cycling until all of the antibodies have been imaged. This is the panel which I was involved in the development, validation and running of as part of my PhD thesis. Obviously won't go through every marker, but it's just an example of the high dimensionality and high plexing that we were capable of doing, including some EBV proteins, which were the latent membrane protein 1, that oncogenic protein I spoke about, and other viral proteins like EBNA1 and EBNA2. These are some of the images that we generate from the phenocycler. I thought the one on the left-hand side really fit in with the colour template of the slides, so I'll put that in there. And then that's some examples of the viral proteins, which the reason that this is that we were so happy to be able to stain for viral proteins because the antibodies are really difficult to work with in general. And the codex chemistry is only compatible with about 60% of antibodies overall. So the fact that we were able to stain for viral proteins was a nice surprise for us. I think, especially hearing people wanting to get into the spatial space, it's really brilliant that UL and Limerick have facilities like the phenocycler that we can use, but I think people kind of forget about once you have these images, well, what are you going to do with them? So I thought it was kind of important to highlight that once you do generate these images, there is a very niche and I wouldn't say difficult, but you have to learn, a process that you have to learn and be able to get useful information from these images. And I'm lucky that in the lab that I was in, Dr. Éanna Fennell, my colleague, had developed 2 pipelines to develop to process the multi-omics data sets. And while there are a good few like pre-processing pipelines available, because proteomics has much fewer markers compared to like transcriptomics, these are quite specific to the processing that I was going to undertake. So as I said, one of the main things that I wanted to do was look at how EBV interacts with the immune microenvironment. And to do that, the first thing that I did was conducted a neighbourhood analysis. And what this did was it allows you to identify neighbourhoods, which you can see as colours on the left. And within these neighbourhoods, what you have is you have populations of cells that co-localise together. So every time that I clustered samples using a neighbourhood analysis, I saw that LMP1 positive B cells consistently co-located with CD68 macrophages. And when I looked at these CD68 macrophages in more detail, I saw that they specifically express 2 molecules, PD-L1 and IDO1, which by different mechanisms cause immunosuppression in the immune system. And as you can see on the staining on the right-hand side there, LMP1 is in yellow and PD-L1 is in pink on the left and IDO1 is in pink on the right. And you can see that the yellow and the pink co-locate, showing that LMP1 was driving expression of PD-L1 and IDO1 on those macrophages, or else recruiting those macrophages. And to prove that this was specific to the LMP1 microenvironment, I calculated the abundance of these immunosuppressive macrophages in the microenvironment of LMP1 positive cells and LMP1 negative cells, showing that the blue line was consistently higher, meaning that these macrophages were specific to the microenvironment of LMP1 positive cells. And to show that this was specific to primary EBV infection, I made use of 2 data sets in our lab of classic Hodgkin's lymphoma and DLBCL, 2 EBV-driven malignancies. And I showed that when I looked at the expression of IDO1 on CD68 macrophages and also on CD68, CD163 positive macrophages, there was a significant difference in the amount of IDO1 expressed on macrophages in primary infection. And there's a few papers that have characterized the role of IDO1 on macrophages. And overall, they report that IDO1 leads to tolerance, immune tolerance, suggesting that IDO1 might be used during primary EBV infection to prevent the killing of EBV-infected cells to establish a latent infection. And I also was able to show that this was specific to the microenvironment of not just EBV-infected cells, but LMP1-infected cells by comparing the abundance of these macrophages in the microenvironment of LMP1-positive cells across the 3 disease conditions. we all know that T cells are really important in immune response, especially for viral infections. So I also wanted to see whether the abundance of T cells was consistent across the microenvironment of LMP1 positive B cells in the benign condition and the malignant conditions. And with the exception of EBNA1, which is on the left-hand column, the EBV negative B cells, which are in red, had consistently higher levels of different T cell subsets. So CD4 in the top row, CD8 in the middle row, and Treg cells on the bottom, suggesting that there might be some mechanism of T cell exclusion by these EBV positive cells. And again, I was able to show that the localization of T cells in relation to LMP1 positive cells was not consistent across benign and malignant conditions. So in conclusion, I was able to use the phenocycler fusion to document the microenvironment in EBV-driven diseases. We show that the expression of IDO1 may be necessary for the tolerance of EBV-infected cells, specifically LMP1-infected cells in primary infection, and that T cells are selectively excluded from the microenvironment of EBV-infected cells during primary infection. And while my PhD didn't look at the mechanisms of these exclusion or how LMP1 or EBV may be driving those exclusion processes, we have now as a lab generated a data set on the same tissues, so IM, Hodgkin, DLBCL, which looks at 1,000-plex transcriptomics. And this is going to allow us to look at whether these the localization of these immune cells is cytokine or chemokine driven, and that's kind of what I'm going to be doing while I find out what I want to do with the rest of my life. So thank you for listening.
Okay, so I think you've answered two of the questions I had, but so the IDO1 is expressed in primary EBV, but not in EBV-associated malignancies.
So only of the two that we looked at. We just looked at Hodgkin and DLBCL, which are both B-cell malignancies, which have similar patterns of EBV gene expression compared to primary infection, which is why we looked at them. And we showed that while IDO1 it wasn't completely absent in the malignancies, the percentage of macrophages which expressed it in IM tonsil versus the malignancies, IDO1 was much more present in the benign conditions.
Okay. And how many cases did you look at in the malignant?
So in the malignant of the Hodgkin, we had, I think, 21 EBV positive Hodgkin and 16 EBV positive DLBCL. Of the IM tonsil, we had less because we found it much harder to get. We only had 8.
Okay. You're going to look at that more extensively now with your data. Okay. And your plans for the rest of your life? You're going to be here for a while with this?
Well, if my supervisor had his way, I'll definitely be here for a while, but yeah, I haven't decided yet, but I'd like to stay in the field
the rest of your life, so we'll give you a chance. Okay, thank you very much. And now it's my pleasure to ask Dr. Máire Lavelle, our consultant breast pathologist at UHL, to speak next. Thank you, Máire.
So thank you very much for that, Ruth. So my name is Máire Lavelle. I'm a consultant pathologist with a special interest in breast in University Hospital Limerick. And what I want to talk about today is something I'm very interested in, which is expanding the role of the pathologist, or my role, in patient-centred care. So the medical landscape is changing and patients are becoming far more involved in their care, and it's becoming a model where the patient is in the middle and then we have the multidisciplinary teams around and care is individualised to the patient. And what I'm proposing, I'd like to talk about and explore, is taking the pathologist out of the laboratory where we normally live and push us back in front of the patients. So I suppose to explain how I would propose to expand the role of the pathologist, I think it'd be helpful if I told you what a pathologist did. So what does a pathologist do? Well, basically, pathology is a study of disease, and as a pathologist, I diagnose breast disease, and usually breast cancer, under the microscope. And I do that by, the tissue is taken from the patient here, and then it goes through a number of, first of all, we dissect it, it goes through a number of chemical processes, it's fixed, and then it's embedded into wax blocks here, and then it is cut into very, very, very thin slices so that light will be able to pass through it. It's still transparent at this stage, so it's dyed, and then it is turned into a slide that looks like this. This is a H&E stain, which is kind of purples, pinks, and blues. And at this stage, I can have a look at this under the microscope and make a diagnosis. So what that means is that when the slide comes to me, I am hiding in this office here, just in case anyone's looking for me, very professional. And I sit at a microscope and I look at these slides and I report them and I put my report onto the system, but I don't actually see the patient. I handle their tissue, but I never meet a patient. So as a result of this, us pathologists, we get a little bit of a reputation. I particularly like this article in the very magically named The Pathologist. Most people have heard the stereotypes about pathologists. They're an unfriendly lot who chose their careers because they didn't like patients or indeed people at all, preferring to spend their time in the company of corpses and assorted taxidermy. And pathologists don't want to talk to anyone, lack a sense of humour, and spend ample time off watching Quincy and Silent Witness. And that hurt because I really do like Silent Witness. In any case, it's a little bit unjustified. I think we're a lot friendlier than people give us credit for. So just in terms of, we'll just talk about the relationship that exists at the moment between patient, pathologist and the treating clinician. So the dark lines is kind of, we're going to say, definite open lines of communication. So the patient talks to the treating clinician and the treating clinician talks to the patient, and they have a two-way communication here. But the pathologist has a line of communication to the clinician in that we issue a report with the diagnosis, and that goes to the clinician. And the clinician may or may not come back to us at all, and we may or may not talk to the clinician. To be fair, in breast cancer, we actually have, and in UHL, we have excellent communication, and they all have me on speed dial, so that isn't really the case here. But in a lot of centres, especially in centralised centres, the pathologist might not even ever meet the clinician. And the patient may or may not decide they want a copy of that report. So usually there may or may not be any sort of communication here. So why does this matter? Because things are changing. So patients aren't these passive recipients of care anymore, and they are actually at the centre of their own care. And particularly with places like in the US, where we have a lot of electronic health records, patients are accessing their information and their reports at home on their own laptops. We also have a multidisciplinary approach to patient care. So multiple teams involved, and some of those teams might not necessarily know a lot or have a lot of expertise in pathology, but they're the teams that the patient is seeing. So if a patient is seeing their GP or seeing a breast care nurse and saying, but what does this report mean? It makes their lives very difficult as reports are becoming more and more complex, longer, more adjuvant tests, molecular testing. And maybe it would be better if a pathologist explained this to the patient. So what are the challenges that we have? So there's an international trend towards centralisation and consolidation of pathology services. Basically, you just have one giant lab serving multiple hospitals. And I did this, I worked in Barts Health in UK in London, and we were one giant laboratory of all laboratory services in one building, and we served 5 hospitals across London. And there's some positives of that because you have a whole load of pathologists and a lot of expertise. Negatives are sometimes it's very difficult to talk to your colleagues, and we certainly weren't seeing any patients. Also, pathologists, there's international shortage of pathologists, increased workloads, pressure at increasing our turnaround times. So you lack the manpower to have these clinics. In spite of all this, in 2008, a bunch of very eminent pathologists here. So this guy, if you see a giant tome pathologist's office, is probably written by this guy, Rosai. And this guy, Epstein, is very well known in genitourinary pathology, and he very suddenly resigned from Johns Hopkins earlier this year, and if you want to know why, I suggest you Google it. But anyway, they all met in 2008 in Sirmione in Italy, and they decided that direct communication between patients and pathologists was a serious need. But out of having this fantastic meeting over a week in Italy, really all that came out of it in a practical sense was they created frequently asked question initiative, Understanding Your Surgical Pathology Report. And that is just a website with kind of frequently asked questions, and it's still maintained by the American Cancer Society. But they created this model of pathology explanation clinics where you have direct communication between the pathologist and the patient, so that we can discuss their diagnosis with them and answer their own questions. But it never really took off anywhere. So what are the pathology explanation clinics? They're a type of care model that provides an opportunity for patients and pathologists to meet face-to-face and for patients to learn about their diagnosis and look at their own histology to see their cancers. So they make pathology visible to the public in a new way, because I think probably for a lot of the public, the sample goes to the lab, we don't really know what happens, and then they get a result. It just could go into a machine for all they know. Also to provide full transparency of the diagnostic process and improve patient understanding of sometimes when we make a diagnosis, it may not be cut and dry. It might be you have a differential diagnosis, and this is why we can't tell you it's definitely benign or definitely malignant. And it contributes to the patient's ability to make decisions on their care. So I just want to give you a taster of what a pathology explanation clinic might look like. So this is me in my office. So I have my microscope here, and joined up to the microscope, this is a camera, and this camera will project onto the laptop live what I am seeing here. Alternatively, this is a teaching arm, so this is another head of the microscope. So anyone looking in here can see what I'm seeing. Although I have to say, if I was going to show it to a patient, I'd rather put it on the screen, so it's just easier to point and for the patient to see rather than adjusting eyepieces. So again, just in terms of, I don't really know the background of everyone here, so just so you understand what I'm talking about, we'll just go through anatomy of a cell. Right? So this is your cell, this is the membrane here, this is the nucleus, and all this stuff here is the cytoplasm, and this all becomes relevant in a minute. So a patient might come into me in my imaginary clinic, and they have had a left breast biopsy and a left axilla biopsy because they have a mass in their breast and their axilla. So this is the report that they would get if they asked for the report, and this is the report the clinician gets. Invasive ductal carcinoma, it expresses estrogen and progesterone, and HER2 immunohistochemistry is equivocal, but HER2 is amplified. And the left axilla biopsy shows metastatic carcinoma. So maybe they have some questions about this. So here is the H&E stain of the cancer. And this is invasive ductal carcinoma here. You can probably see these cells. This is a nucleus, and the cytoplasm is just around it, is paler. These nuclei are all different shapes and sizes with angles. These are benign cells here, so you can probably appreciate the size difference between more of a benign cell and a malignant cell here. And this is why this is a grade 3. Grade 3 is the highest grade you can give a cancer. It doesn't express any tubules, it's poorly differentiated, and has a lot of mitoses being cells dividing, and you can just see one up there. It expresses estrogen and progesterone, so this is immunohistochemistry. So if this is basically, we put antibodies on top of the tumour cells, and if they express the antigen, so estrogen and progesterone, they turn brown. So this is strongly expressing estrogen and progesterone, and that's important for the patient's treatment. They might have anti-estrogen treatments. E-cadherin is positive, and this basically is diagnostic that this is a ductal carcinoma as opposed to other types of breast carcinoma. And this immunohistochemistry is actually staining the membrane as opposed to the nucleus, which was staining the previous. And this is HER2, and HER2 is important because of Herceptin, and that's kind of, so in the last couple of decades, the miracle drug that has, revolutionized breast cancer. And we do an immunostain, and if it expresses Herceptin, the patient can get this Herceptin treatment, and if it doesn't express it, then the tumour is not going to react to Herceptin. In this case, so if it's strong, it should be very, very strongly encircling the cell, but the stain is equivocal, so this is what we call 2+ staining. So it's not positive, it's not negative. So we have another test, and this stains up the chromosome, the HER2 chromosomes, in relation to the chromosome 17 within each cell. So this is one cell here, and this is called D-dish testing, or in situ hybridization testing. And this, I can tell this is amplified. So the black is the HER2 gene, and the red is the chromosome 17 gene. And you can see in this one cell, there's about, 12 compared to 2. Normal would be 2 and 2, so this is clearly amplified. A little bit more complicated than that, but we'll just keep it simple for now. So this is their lymph node. So this is normal nodal tissue here, and this is the cancer invading the lymph node. So this person has cancer in their lymph nodes. It is metastatic. So just go back to our initial model here, where we don't really have much communication unless the patient gets the report, and we may or may not have communication just in the form of a report with the clinician. Hopefully to change that to, we have two-way communication between the pathologist and the patient with the PEC. And in addition to a pathology report, the clinician will also get a note in the chart. I still like to think that in Limerick, definitely this line is very, very black. You could actually have 3 lines or 4 lines down here, but we definitely communicate quite a bit. So why aren't we doing this? What are the obstacles? So main ones, buy-in from the key decision makers in the clinical pathology departments, also resourcing. As I said, there's a worldwide shortage of pathologists. Would be better off diagnosing more cases and having better turnaround times than investing all this extra time. But the benefits for pathologists, if our role is more visible to the patient, it may help with recruitment and resourcing of our services. What happens when you're in the laboratories, because the patients don't see you, it's often a prime place where cutbacks are introduced because, well, we can just shove it all together and consolidate it, and the patients don't know if you don't have somewhere to report, because they don't see it, but they will see clinics. They will see, waiting times in the clinics decreasing. They're not going to see pathologists. Also, if we have clinics, we could hopefully streamline the role of the clinicians so they don't have to worry about explaining all the diagnostic part of the patient's cancer and optimising their appointment times, and also empowering patients. Just let them, if they know more, they might want to make different decisions. If they actually physically see the cancer, they might feel differently about maybe having more tissue taken out if a margin is positive. So how do clinicians feel about this? So basically clinicians, 83% are in favour, and the other 17% read that earlier pathologist article and thought that pathologists generally shouldn't be in front of patients. So there was a very, very small study done. It was published this year, and it was a prostate clinic. And they found that they did interviews with patients and also with their treating oncologists with the pathology explanation clinic. And the results were that there was an improved patient understanding of their diagnosis, it supported them in their treatment decisions, and the participating oncologists noted improved understanding and decreased patient anxiety when they were more educated about their diagnosis. So what about UHL? What do we have? Well, as you just saw from the photograph I showed, we have the equipment. It can be done. And because as well we have that digital camera hooked up to the laptop, that means that I could possibly do these clinics remotely. I don't necessarily have to physically have a patient in the room. And I think we have buy-in from the clinical and pathology departments. But what we lack is manpower. I think this would be a fantastic project or fantastic way for trainees to see patients and to see why they're doing their job, why their job in pathology matters, why the diagnosis matters. So I unfortunately have no trainees, and I'm a one-woman show at the moment, but I'm hoping to introduce this as a project, and I have some lovely medical students from UL who have kindly volunteered their summers. I don't know why. But I'm very grateful, actually, that some medical students are going to participate. So hopefully we can do a pilot project and get this off the ground. So thank you all very much.
Just one question. Okay, Tofil.
Thank you, Máire. Excellent presentation. I'm on the opposite side of this siloed pathologist. I'm a physicist and I'm the head of physics, and we have the same problem. So we have the opposite triangle that you have, a lot of innovation in molecular and chemical pathology, but we have no link with the clinicians, the decision maker or the histopathologist. Okay, so how do you see that innovation triangle be merged with your patient-oriented the triangle that you showed? How can we merge that?
Just summarize merge in terms of research scientists with. No, I think it isn't a lack of willingness. I think, to be fair, on the clinician's part for the most part. I think where we fall down is that even though clinicians should, or histopathologists, and when I say clinicians, I mean histopathologists, all doctors, we should have some protected time for research. We should have protected time for these things, and we simply don't, and the service just takes over. And I think in the UK, I've worked in the UK, and they are better at giving that protected time to allow clinicians to invest more in research. But I also think that days like this, where certainly as someone here referred to it as speed dating among researchers, and I thought that was very good, but days like this where you come together and actually meet each other and network and see who's interested in what, and who can assist and help. Certainly my door is always open, always contactable, and I'm happy to assist anyone if they're looking for a project. But I think there are this is a very long answer, sorry, but I think there are other multiple ways that we can maybe establish this, especially between a university and a hospital. And I think one of the ways is when we had that SWOT meeting, and I stressed at the time is what we need is a list of contacts of people and what they do. Are they open to research? Are they open to, and have a directory, so that if you're looking for a histopathologist, you'll see my name and you'll contact me and go, can you help me with this project? And I'll go, hopefully yes, or no, I'm not interested. No, but I mean, hopefully yes. But I thought that would be, I said I've only started in Limerick, I've only been working in Limerick for a year, so I'm still meeting people. That's why I enjoy coming to these meetings, so I meet more people and network and for people to get to know me. But I think that would be one way to, another way that might be helpful to get everyone to link together.
Can I just I'm just laughing here because I'm looking at Tricia because I actually just said to her we sort out the directory today. So on that, I just want Tricia, can we introduce you? So hopefully Tricia will be in contact with lots of people here within the coming months and year as we kind of actually put that directory together.
Thank you very much, Kieran. If we have discussion, we'll come
Tricia English, the new senior administrator.
Any other questions we can just come to at the panel discussion. So finally, I'd like to introduce Dr. Amira Mahdi, who's going to speak to us about breast cancer again in the context of the tumour microenvironment. Thank you very much, Amira.
So, thank you very much for the opportunity to talk here today, and it's been lovely to see everyone's talks so far. So I'm going to talk to you today about my research into advancing therapies for the treatment of breast cancer. And we can really think of breast cancer treatment as one of cancer research's success stories, because it was one of the first cancer treatments the first cancer types where we understood what exactly was driving the growth of the cancer, and we can target it specifically and then spare patients from the harsh side effects of high-dose chemotherapy. And prognosis for early-stage breast cancer at the moment is really good. However, we do still face some challenges when we're trying to advance therapy. First of all, we have when cancer spreads around the body in metastasis. And for example, when cancer spreads to the brain, a lot of the drugs we have that work really well in the rest of the body can actually struggle to reach the brain, and making the brain tumours much harder to treat. Another big challenge we have is that of resistance. So as we're developing more and more of these advanced targeting therapies, cancer cells can evolve and develop mechanisms of resistance against these therapies. So despite an effective initial treatment, over time, cancer cells can develop resistance and can recur in causing secondary or metastatic breast cancer. And then, of course, we have the influence of the tumour microenvironments. We've talked a bit about this already today. And we now know that when we're developing these new cancer treatments, we can't just think of the tumour cells in isolation. We need to think about their environment. This is really important when we think about therapies such as immunotherapy, and that actually relies on the action of the tumour microenvironment for it to be successful. So I'm going to talk a bit today about overcoming this challenge of resistance. And this work was started off during my postdoc in Dublin City University. And we were working with a company called Puma Biotechnology to identify new combinations for their tyrosine kinase inhibitor drug called neratinib. Neratinib is approved for the early stage and late stage treatment of HER2-positive breast cancer, which Dr. Lavelle was mentioning. And we have a lot of drugs that can target HER2 on the cells when we have HER2-positive breast cancer. One of these is called trastuzumab. This is an antibody that targets HER2 on the surface of the cell. It's also called Herceptin. And as I mentioned, we have tyrosine kinase inhibitor drugs like neratinib, which target HER2 intracellularly. And the discovery and approval of drugs such as neratinib were really revolutionary in HER2-positive breast cancer because they transformed it from a poor prognosis cancer to one with quite good survival rates. However, for a large proportion of patients, we do know that over time they can acquire therapeutic resistance to these anti-HER2 therapies. And when I was looking into this, a trend that started to emerge was that the action of the estrogen receptor pathway could be a compensatory signaling pathway that allows the cells to acquire this resistance.
And the reason is really when we look inside the cell, it's an area of really complex crosstalk between the pathway of the estrogen receptor and the pathway of HER2. And the cells can kind of use this compensatory pathway to then develop these mechanisms of resistance. And interestingly, like we saw this in Dr. Lavelle's talk as well, it was really good that you were before me, quite a large proportion of HER2 positive breast cancers are also estrogen receptor positive. So this overlap exists even prior to the patients receiving therapy. And because of the compensatory signaling between these two pathways, there's a really strong rationale for the dual targeting of both HER2 and the estrogen receptor in the clinic. However, it can be quite an underutilized strategy when considering treatment for HER2-positive metastatic breast cancer. A lot of the treatment guidelines may go with chemotherapy type regimes first. So at the moment, there's a bit of a lack of evidence for it to be used widely within the clinic. So one of the first things we did was look into the publicly available data for patients within this cohort. And we saw that there were patients who were HER2 positive and estrogen receptor positive who were not getting this hormonal therapy. And in terms of how this relates to patient outcomes, we saw that the patients in this cohort that did get the hormonal therapy did see benefits in things like relapse-free survival. So one of the limitations in the use of this strategy may be because the drugs we have are a bit limited. So when clinicians are treating advanced ER-positive disease, one of the drugs they use are called selective estrogen receptor degraders, or SERDs, and these break down the excess estrogen receptors in the cells. There's been a bit of a standstill in the development of these types of therapies. So for over 20 years, fulvestrant was the only available approved SERD. This drug works well, but it's very limited, so it has very poor bioavailability, and it leads to very lessened efficacy in vivo. It also has to be administered by quite uncomfortable and regular intramuscular injections to patients. So for the past 2 to 3 years, a lot of the pharmaceutical companies, there's been a big push to develop orally available SERDs that have a better pharmacokinetic profile than fulvestrant and can be taken in a tablet form. For example, elacestrant, which was approved in early 2023. Apart from this one approval, we haven't as of yet seen the true potential and the widespread benefit of these next generation endocrine therapies. So we were thinking the benefit may lie in combination therapy. So we wanted to see whether neratinib and these orally available SERDs could be a synergistic drug combination for the treatment of HER2-positive, ER-positive breast cancer. And for the sake of time, I'm just going to talk about one of these SERDs today, and it's called camizestrant. This is being developed by AstraZeneca, and it's currently in early stage and late stage ER-positive breast cancer phase 3 trials. And to do this, we used HER2-positive, estrogen receptor-positive cell lines, and we conducted what are called matrix assays. And in these, we treat the cells with ascending concentrations of drug 1, in which case this was neratinib, and drug 2, which was our oral SERDs. And you can see from the analysis here, we basically use a software that can compute what were the interactions between these two drugs of interest. So in the lower grids here, blue represents a statistically significant synergistic interaction between the two drugs, green represents additivity between the two drugs, and yellow to red would represent antagonism. So as you can see, we have a lot of blue. The SERDs and neratinib were working really well. And then of the 5 different combinations we looked at, camizestrant came out with our greatest maximum low synergy score. We brought this forward to a bit more testing and using this nice system where we can image the cells in real time, and using a fluorescent caspase-37 dye, we can look at the cellular apoptosis. And we saw that the combination of neratinib and fulvestrant, which is our older or standard of care SERD, and neratinib and camizestrant caused the greatest increase in cellular apoptosis at 72 hours. We then looked at the cells in 3D cell culture. So in this, we grew the cells as tumour spheroids, just to kind of represent a bit better how the cancer cells would exist within the body. And again, we saw the potential, I suppose, of the neratinib and camizestrant treatment. As you can see, it showed the most significant effect at about 96 and 120 hours in reducing that tumour spheroid growth. Just a few more cell lines. We looked at some other models of breast cancer. So for example, we have an estrogen receptor positive NRG fusion mutation. So this cell line has a mutation in uregulin, which is a ligand for the HER family signaling. And again, we saw synergy displayed by the blue colouring within the cell lines. And we also looked at CAMA1. This is a estrogen receptor positive, but it's a HER2 low cell line. So usually it's a model of a subtype who wouldn't usually get these anti-HER2 therapies, but we were seeing synergy between these two drugs in this cell line. We also looked at a model of acquired resistance to trastuzumab and pertuzumab, and these would be considered first-line therapies for HER2-positive breast cancer. And these cell lines are developed by exposing the cell lines over time to ascending concentrations of the drugs until the cell lines can kind of grow and proliferate with the drugs in their culture media. And interestingly, we saw for this combination that the two resistant cell lines actually showed greater synergy than the parental or sensitive cell line, despite very little difference in the sensitivity to the neratinib drug alone in these cell lines. We looked at the signaling, so what was happening within the cells when we treated them with these drugs. And one of the things we saw is that when we use neratinib alone, so that's the anti-HER2 targeting agent, we see this big increase in the expression of the estrogen receptor. Then when we bring in back our estrogen receptor therapies, like fulvestrant and like camizestrant, we knock that signaling way back down. We saw this in vivo as well, so this is some really nice staining work that was done by one of our biomedical science final year students, Eilish. And we saw that in mouse xenograft tumours where they were treated with neratinib for 4 weeks, again, we see an increase in that expression of the estrogen receptor. We looked at this on a gene level, and this is important when we're thinking about estrogen receptor signaling because the estrogen receptor acts as a transcription factor. So we looked at some of the targets of the estrogen receptor, such as PGR, progesterone receptor, and GRB1. And we saw the same sort of trend where when we have that single agent treatment, neratinib alone, we see a big increase in the expression of these associated genes. And then this is knocked back down when we include our SERD drugs, our anti-estrogen drugs. This is not just limited to in vitro cell lines. We also saw it in patient samples. So we looked at patient samples from the TRIO-B7 trial. And in this, patients' biopsies were taken pre-treatment and then also 2 weeks post-treatment with anti-HER2 therapy. And again, we see this trend where increasing expression of estrogen receptor-associated genes. And if we look a bit closely at patient outcome in this area as well, we saw that for ESR1, which codes for the estrogen receptor, and CCND1, which codes for the protein CDK1, that the levels of gene expression of these genes were actually higher in patients who had residual disease post their neoadjuvant therapy course than the people who experienced pathological complete response or no tumour was found at the end of their neoadjuvant course. So again, that's linking this compensatory ER signalling to outcomes in terms of people who may have resistance to therapy and people who may have residual disease after their anti-HER2 treatment. So in conclusion from this work, we saw that the combination of anti-HER2 TKIs like neratinib and the orally available SERDs like camizestrant are very synergistic in these preclinical models of ER-positive, HER2-positive breast cancer and shows potential in being able to overcome that therapeutic resistance we see that's very driven by estrogen receptor signaling. We think there's strong evidence for the further exploration of this combination at clinic because of the synergy we saw, because of the ability to overcome resistance. And also when we think about the patient factors in this, so the fact that this is an orally administered drug, it may be easier to take for the patient, and we saw it was well tolerated in vivo. So where is this work going? So in terms of this next generation of hormonal therapies, I really think more work is needed to understand where they can kind of lie within the clinic and for patients. So although we've had one approval so far, there's been a bit of a difficulty in getting these drugs past clinical trial and approval. So one of the problems is the sheer volume of drug candidates. So you can kind of see from this list here, we have a lot. It's a very popular area for pharmaceutical companies, and being able to choose the best ones to bring forward to clinical trial is quite difficult. We also have the clinical trials that are carried out. They're also often carried out in patients who have had multiple lines of prior therapy. So this is something we need to take into account. You know, have they got CDK4/6 inhibitors previously? Have they had aromatase inhibitors previously? Combination strategies is also really important. So
it looks like really that these drugs as single agents are kind of struggling to beat the current standard of care. So their true potential might lie in being combined with other therapies, like as the work that I've showed you here today. And then finally, so we want to take into account the tumour microenvironment. So we've talked a bit about this today, and this is an example of breast cancer tissue. And we can see that a very large proportion of the tissue in the breast is made up of adipose tissue or fat cells. And we know that fat cells are really important in both the synthesis and the regulation of hormones, and the interplay between the fat cells and the tumour cells can alter treatment response, both in vitro and in vivo. So we want to take all these factors into account when we're trying to figure out which are the best candidates from these next generation hormonal therapies to bring forward to clinical trial. And so I'm very grateful to recently receive funding from the Health Research Institute here at UL to do just this, so in collaboration with Professor Kieran McGourty. So we want to make co-culture models of breast cancer cells with adipocytes to try and understand how, when we incorporate in the tumour microenvironment in this way, can we understand better how this next generation of hormonal therapies can respond. We also want to see whether the cancer cells and the adipocyte cells themselves interact with each other in reciprocal reprogramming. And we also want to incorporate in models of acquired resistance to prior therapies. So very often these drugs get to clinical trial after showing really good results in the lab, and they're met with patients who have had 3-4 lines of therapy. So they're not going to show effect in that case. So we need to be testing that way before we even get to that line of clinical trials. So we want to use these models of resistance and then also work, analyze them in depth using single-cell RNA sequencing to really identify the subpopulations that are driving this resistance and whether we can pharmacologically target these modifications that drive resistance as well. And really, we think that developing these more complex and more physiologically relevant preclinical models of breast cancer are really important for us to make our findings in the lab much more translatable to successful clinical trials and much more relevant to the patients who need these treatments the most. And so with that, I'll just say thank you to everyone who helped with this project so far, and thank you to the LDCRC for organising this very good day.
So if you want to just join us for a brief panel discussion. What time is it actually? Sorry, I don't... Okay, so we're kind of over time, so we'll just do a couple of quick questions. I suppose just hands up if anybody has a question. So for Kieran and Amira, it's very nice to see, you know, some overlap, you know, with the technologies, and I suppose that's the whole point here, isn't it? We're trying to, you know, not just be, I suppose, we're disease agnostic and area of expertise agnostic, so that we're working together. So that's very nice. Kieran, do you want to make any comment on that? I mean, the link between both of your projects, and obviously you're heavily involved. Yeah.
It's very, very, very satisfying to see. We started a few years ago with an empty lab, and I think we have a cutting-edge facility now, supported by industry, supported by graft, a lot of hustle and a lot of graft, and also supported by the clinical interaction we have in UHL. So I think Amira's project is a perfect example of where she's bringing a lot of expertise into our space, and we have an environment in which she can really ask the questions that she wants to ask and avail of maybe some in-house expertise that we could help contribute to your project. And I think it's increasingly becoming a compelling argument what we're building here, and I'm grateful to it, grateful to be here.
Thanks. And Amira, you've been linking in with the breast subgroup that we have our clinical and scientific leads on. So hopefully, I don't know how you're finding that, but hopefully you're getting access, I suppose, to expertise and patients.
So far, so I think it's going to be a great opportunity to, I suppose, make the most of the samples that we do get in, and being able to use them in these kind of models and try and learn a bit more about where drugs could be needed. Like I said, there's just we have so many drugs, so many in many different lines of development, and it's really, really hard to get them across that finish line. So improving the models that we have at early stage is kind of needed.
And I suppose we need national collaboration. We're not going to be setting up a phase 1 unit here in our cancer clinical trials unit. So you have your links with DCU and Professor Crown. So I suppose, is that still active?
Yeah, of course. So we really, once we kind of publish this on our combination, like we really hope that it will take off a bit, and there is a lot of interest in combining these novel orally available therapies with other, CDK4/6 inhibitors, and as well as the HER2 targeted therapies. They're almost becoming this catch-all where we have a lot of HER2 low breast cancers, we have a lot of HER2 low other types of cancers. So it's more like it's becoming more personalized. It doesn't really matter the organ, it's more to do the molecular subtype and what we can target. So it's still hard to do phase 1 trials in Ireland though, but hopefully that'll change.
Yeah, that's where we have to be national. Have I any questions from the audience?
So within kind of the drug repurposing space and sort of like the Droop-like trials that are going on throughout Europe now, is this data, is there like a mechanism to feed, was it to either scale up these kind of grids, and I don't know, this may well be done, I don't know, within more high throughput labs as well, to scale this up and then feed that into the drug repurposing trials?
Yeah, no, certainly. Like I did my test that I showed you there, 96 subplates, that's a certain level of throughput, but it can definitely be scaled up quite a bit. And you can, if you did these high throughput non-biased screens and just combined loads of drugs, I'm sure we'd discover a lot of synergistic combinations. It's important though to, we can use the drugs that we know are safe for patients and we know are well tolerated and we know that people are taking already, and it can kind of make the journey a bit easier than picking some compounds at random. But I think it's definitely something that can be scaled up hugely, yeah.
Thank you. Paul?
Thanks, Ruth. For Christina, I was very struck in your presentation, the upsurge in younger presentations with colorectal cancer. Is there reliable research into what's driving that? If there isn't any conclusions or hypotheses, is the research being done?
So it's kind of the million-dollar question at the moment, and certainly from a biological profile point of view, we are seeing very specific, like aggressive features within patients who develop, let's say, rectal cancer in their 40s compared to somebody who develops it in their 80s. It's environmental interacting with genetics, but we've known that about cancer for a very long time. We just don't know, the specific drivers. Obviously, there's kind of obvious culprits. I mean, obesity is one of them that's being brought to the front, and obviously in breast cancer and endometrial cancer as well. But the honest answer is we don't really know, but we're certainly seeing specific biological profiles that are different between younger patients and older patients, and there's large collaborative data on that globally, and I think you're doing considering looking at some, is it progesterone receptors as well? Eoghan, I think we spoke about looking at the progesterone receptor within those younger patients, or not progesterone, sorry. The perineural invasion. It's all progesterone and estrogen for the last talk. Perineural invasion within that group as well, because there's some small data in that, but biologically, yes, they're absolutely different. Why? We don't know why they're happening.
Week, which I think is going to be out in Nature this week or next week, from folks in the Sanger, that's actually, I think, hopefully going to enlighten some of this. And they actually looked at signatures globally in the mutational signatures. So they actually sequenced DNA and they looked at mutational signatures associated with different cancers worldwide. 1,000 patients in 5 different types of cancer. 4 or 5 different types of cancer, one of which was colorectal cancer. And they're picking up these signatures that seem to be associated with early onset cancers. And they are exploring if that's associated with different diets, but equally with different pathogens. And one of their interests was actually within oral microbacterial impacts that seem to maybe be associated, but I think it's still completely out. But there does seem to be different, like, mutational signatures that might be associated with some of these diseases.
Thanks, Aideen. Any other questions from the audience while I'm waiting for maybe one final question from the audience? Máire, your clinic with pathologists, I'm trying to picture it. So I really think into the future it is something that might happen, and very much from, say, our biobanking group that we have. I keep plugging our little subgroups that we have in the LDCRC. You know, patients will really come on side if they can, you know, see an image, be explained, you know, exactly what's going on there. And then, you know, our patients are quite keen to give us samples and be involved in research. But I think, you know, bringing all of that together, it would work very well. We're talking about our patients coming into UHL. It's a cancer centre, so research is just implicit, and that would be part of it, I suppose. That's how I'd visualise it as well. Do you want to give us a little bit more?
Well, I suppose there's a few ways you could go about it. I think it's become a hot topic basically at the moment, bringing pathologists out and bring them in front of patients. So we had the International Pathology Day last year in RCPI in Dublin, and the entire talk was basically about this and about making pathologists more involved, direct involvement with patients. As I said, I think what I was hoping to do in Limerick is to start small and do a small pilot project. And one way of doing it is to do it in clinics such as haematology, where a haematologist is looking at slides as well, or in dermatology, where they have some, you know, histopathology training as well. And that would be another way that we can expand our numbers, because I'm limited on manpower to do it. The other thing I was thinking of was piggybacking. Even though I said this is something you could possibly do remotely, I don't think I'd have I suppose even the clerical in terms of getting the charts and so on, everything would be very difficult logistically to start out like that. It'd be easier for me to go to a breast clinic and then approach patients there, you know, and say, do you want to, and then get their response. But that's probably how I would, you know, I would start small and go about it that way.
Yeah, and I think there's such issues with, you know, recruitment of histopathologists. That's not going to get better. So the job needs to change somehow. And I think exactly bringing people out of labs to meet people, as we can see today, bringing us together to meet each other does really help. So,
well, I mean, to be fair, I think most of the public, when they hear pathologists, they think CSI, bodies, murder, and that's fair. That's the public perception. Like, we are. I'm sure if you ask most people on the street what happens, you get a biopsy. What happens to it? And they go, well, I get a result, but nobody, you know, and that's because we're hidden away. But maybe we should get more people interested in it. And I mean, I do find I get a lot of transition year students and so on, and they just go, we had no idea what it was. And yeah, you just need to, and in terms of getting resources and, you know, attracting people into the specialty, we need to be out there more, and positive impressions rather than the one that I showed in my presentation.
Getting SPRs to Limerick, I think, is the first step.
Definitely.
Any other
Just
comments?
one more question. On the combination therapy you spoke about, I'm just referencing previous knowledge from a project pilot we did for HER2-positive breast cancer patients and using Herceptin by Roche, yeah? So it was standard practice that for the first 7 cycles, I remember the clinical recommended was 18 cycles of Herceptin. So for the first 7 cycles, they had a combination of drugs. They had Herceptin and doxycycline. And I think that was by Sanofi. So I'm just asking, is that part of the kind of combination therapy that patients...
So that combination is a combination of both targeted therapy, which is the Herceptin, and chemotherapy, which would be the docetaxel. So I think for a more, I'm not an oncologist, but I think for a more advanced disease, you would incorporate in that chemotherapy because it's more aggressive disease. You want to hit it harder and get rid of as much of the tumor as possible. But when you incorporate these chemotherapies, like we've studied them for decades and decades, but they do have harsher side effects, such as, you know, your hair loss, all your classical side effects that you associate with chemotherapy. So when we're thinking about these combination targeted regimes, one of the benefits is that we may be able to omit chemotherapy from this. So we may be able to give people much more precise and targeted treatments, less broad chemotherapy, hopefully less side effects. So yeah, that's the combinations that I was looking at.
So final question to Tofil, and then we'll wrap it up.
Thanks. Again, it's addressed to Máire. It's funny that you were talking about additional manpower. So I'm just, without the focusing on the word man, I was just thinking, when you said that, did you mean that there should be more human resources to the hospital with clinical expertise, or do you see any opportunities for who are not medically trained, like not a pathologist, but working, contributing towards pathology, like a chemist or a biochemist or a physicist?
Yes, so please submit your CV to. No, it's very good that it's interesting you brought that up. So when I was talking about that meeting in Ceremony, I obviously only touched on it briefly. It's had 15 minutes. I'm sure I went way over time. Apologies. But that was one of the things that they spoke about during that meeting, was actually developing a role for, I suppose, they're not necessarily medical trained, but I think they call them pathology assistants, who would deliver these clinics, so that they wouldn't necessarily be delivered by consultants, but would be delivered by someone with training, and that anyone doing these clinics should have training. So they did actually create a potential role, a whole new role for the hospital. Just practically from my point of view, it's more that I'm just lacking, you know, pathologists or trainees. If I had a couple of junior doctors, this would be, I think, ideal for them, and I think it would contribute more to their training than maybe, you know, constant dissecting, or I'm sure they get lots of that during their training. And also, they wouldn't have the opportunity to do anything like this in any other hospital in Ireland. Now, during my training, I did go to clinics, to breast clinics, so I went into radiology and I went into the clinics. But sometimes, particularly in centralised labs, like in London, the trainees do not see the patients at all. In fact, they barely even see clinicians. And I don't think, excuse the pun, I don't think that's healthy. I don't think that's good. I don't think it's good to just reduce everything down to a slide. And I think you can see some of them start forgetting this is actually a person, not a piece of glass or a specimen. So for what I meant by manpower for me was more just, I just don't have, you know, pathologists or trainees to help me. But yes, in the meeting, they went through creating a whole new role for that, maybe for physicists as well.
Yeah, I suppose sometimes a report, there can be an element of subjectivity from the pathologist.
Oh, yes.
You know, and I suppose, do you think actually that would impact, like, you know, having that face-to-face patient interaction would have an impact on your kind of, I guess, your view and your attitude towards the actual, you know, the final report, because you're seeing it far more in context than actually, because so much of it is subjective. We think it's like black and white. It's so not. Like, you know, we can get that but it probably takes a long time, like as a clinician, to get to that point where you see it from the other side.
Yeah, I think that's a misconception about pathology, is that it's paper matching, that you just look at it and it's benign or it's malignant, but actually it's really interpretative. So, you know, you have to take things in context, and sometimes it's very difficult, or your differential diagnosis might be a benign and malignant entity. And yes, there is subjectivity, and sometimes you'd say, well, in the clinical context, I would favour a diagnosis of. And sometimes we don't have the clinical context, because we get a specimen and it's just labelled colon. Yeah. The end.
Apologies.
And you know, so I think what I'd say is, if you get very few clinical details, your answer isn't. So the more clinical details you have, the better an answer you will give. No, I agree with you, and I'd hope as well for the patients that if there is, I suppose, some uncertainty, or I've had several cases where, they're just very difficult, and ended up going to the UK and then to the US. But to be able to say to the patient, well, this is why this is struggling, this is why we're sending it to an expert internationally, and we are sorry for the delay, but there's a very good reason, because if we can't make this clear, we don't know what your treatment is going to be. So I hope it helped patients in terms of they're not just at home kind of worrying, going, why is it taking 3 weeks for my result to come back? I suppose we're, at least with clinicians, we have MDTs that hopefully we can talk about these things. But really in a pathology report, again, we have to just, I think pathologists, we need to be cognizant of that it's not just about this is the report and what I think, it's about what is going to be done with that report. So we have to be practical as well. And I've seen plenty of pathologists who just aren't, but we're all very different.
Thank you very much. So I'm just going to finish up, and I think, Tofil, you want to meet some patients. So we're hoping that after the summer, we're going to have a kind of public and patient day. It might be around October. We're working on that. And it would be great, and maybe during that we might sort of showcase maybe some of this, the pathology clinic, maybe Tofil, some of the diagnostics, and something like that might be built into the timetable. So that's after the summer, and you'll hear about that. So I'm going to hand over to Adi now. So just before I do, I want to thank everybody, all of our speakers, and everybody for attending today. I think it has been a great day. We're going to have a meeting in UHL and the CIRC in June. We'll circulate the date in the next week or two. And then, as I said, that kind of patient and public day after the summer. So I'll hand over to Aideen to close now. Thank you very much.
Thank you. Thanks, everybody, so much. I recognise that it's getting late in the day, so I really, really appreciate everybody that's here. Thank you for coming. That's the first thing. The more we get to know, for me, it's really, really important that we build a centre here that's very multidisciplinary, because I think that is one of our unique strengths. And I think just the breadth of different sides of cancer that we have seen today, from surgery to pathology, to actually taking that tissue and staining it like in the spatial biology context, to looking at the single cell side of things, to looking at sensors. That is such an enormous spread across like medicine, biology, physics, engineering, and computer science. We're capturing half the university there. And I think that's what we do need to bring to cancer. 1 in 2 people in Ireland will experience cancer. The number of people that will be diagnosed in Ireland is going to increase because we have an ageing population. And so we need to be a centre that brings all of the heads to the table. So the more we start these really complex cross-disciplinary discussions where we don't know what, I will be the first person to say that there was a whole load of words said in particularly the surgery talk that I'm just going, I have no idea what that word is. I wouldn't even be able to spell it. And that's where we need to be. We need to be in that space where we are uncomfortable with what everybody else is saying, because we're learning. Because we can't be an expert in everything, but the more we learn about what everybody else is doing, the more we can bring something new and innovative to cancer research. And so with that, I want to thank everybody today. Thank you so much.